Yeah. Well, at least good thing I caught it. I know. Whatever. Anyways, we're nearly done right now. Well, not even. Well, kind of. I mean, nearly done with Yankee. So we have we're on 10-minute break. This still has orange juice. I still have to finish this. I'm like really tired right now. I think I'll probably take the B12 when I come back. And yeah, and then let's see. And then I'll calculate how far I really am behind and just see where we go from there. We're back. Let's put on the started bleeding yesterday. Started bleeding yesterday from my nose. It's because the air here is so, well, I guess just the air in my room. It's too dry. You know? Anyways. Whew. What a day you'll be, huh? Let's probably turn on the air filter. Okay. Ah. Oh, maybe put some hydrox after this. Let me see what's up. We got it. Yeah, we'll figure it after. Let's go and take our secret weapon. Oh my god. Oh man, I'm so behind. Let's go. During translation. Oh, vision. Do you need one? Okay. I mean, promote a region. Promote a region at the top box. Gold Yuprata. Helix. GNC. DNC then TA. RNA polymerase. Complementary. Complementary is the coding strand, or the antisense strand, or the template strand. Translate it. Size and on that. That is the non-coding or sense strand. Oh wait, that's the coding. Sorry, that's the coding. Whoops. I'm getting it mixed up. It's the coding strand. The coding strand. RER. Helix. Ribosomes. DNA primase. DNA replication. DNA primase. Oh, wait. Oh, I'm not reading it right. That's the coding strand. In ribosomes and RER. Oh, rough. rRNA. Leading strand. Snowdrop. DNA. Lagging strand. Proteins are synthesized by ribosome. DNA primase. Oh man, I keep focus on trends. Proteins. Exonuclease. Kinetic. Envelope. Two phospholipid bilayers called the inner and outer membranes. Single-stranded. Site of protein synthesis. Well, yeah, translation. Second column. DNA polymerase. Yeah. DNA transcription. Replication. Oh, bubble. Transcription bubble. Transcribed. RNA. Do not. Protein synthesis. $305. Protein modification. Post-translational modification. DNA polymerase mixing enzyme. Condensation reaction. Transcription bubble. Double stranded. RNA. Oh yeah, and the nucleolus. Okay. So that RNA polymerase impacted the reaction during translation. Elongation. Elongation. Coding the non-coding strand. Non-coding strand. That was the strand. Call the promoter region top of the box. Helicase. RNA polymerase. DNA polymerase. Leading strand. Lightning strand. Rough. Helicase. Coding strand. Single strand. Exonuclease. Ocosardi fragments. Second organ. Post-translational DNA polymerase. DNA polymerase. Double stranded. Transcription bubble. RNA poly. DNA poly. RNA nucleotides. Let's reprint the 5' direction. Elongation. Nucleolus. Yeah. Changes in gene expression that do not involve altering DNA sequence. Epigenetics. Histone acylation, DNA methylation is changes in gene expression that do not involve altering the DNA sequence, such as DNA methylation or histone acylation. Yeah. Okay. Yeah. That's pretty good. Jeez. Okay. All right, guys. We finished the Anki today. The Anki was supposed to do when we wake up. Oh my god. All right. I'm gonna just take another 10 before I really get started. Let me finish eating this, drink this, and let's see how I can get some more energy. I'm just feeling quite tired, eh? Mm. The New Yorker, huh? The New Yorker. Let's put you guys on the. I need to just work out for a little bit. And then I think we can do maybe do like 4 hours straight, no breaks. I don't know. All right, let's just take 10 minutes. Let's take 15. Yeah, I need to get like my blood flowing. Let's take 15. Let's do a quick workout. Yeah. I think it's too small, no? And then because of copyright, I'm gonna have to just mute it for a second. Strange. But, okay. Yeah. Sure. Yeah. It is what it is. It looks like we're back, so let's continue. And let's go back. Sure. Yes. Something I have to do too. I need to transport this in memory. Oh, okay. Oh, okay. 4 sections. Okay, so we have to get through 4 sections today, not 3. Plus, we have to read. Plus, we have to read the IFT high yield, and add Anki into Anki tonight. So maybe I can leave this stuff for tomorrow. Some of that stuff I was gonna do. It's okay. It's not like it's like really needed right now, anyways. Let's take out. And really we only need one because I can do one side for each section. Let's take out one section and get to it. I don't know. Yeah. Let me get my book. That one's 40. I do want to actually go back a little bit though into section 7 and just read and just watch. Maybe you can watch this one. I can hear you, lady.
Cell to have the versatility and the adaptability of an efficient ninja, so that it expends energy to express the appropriate proteins only when needed. Or you can think of the cell as a lazy couch potato that wants to expend the least amount of energy as possible. So starting at the beginning of gene expression, let's talk about gene regulation as it pertains to DNA and chromatin regulation. Let's talk about the structure of DNA. DNA is packed into chromosomes in the form of chromatin, also known as supercoiled DNA. And so chromatin is made up of DNA, histone proteins, and non-histone proteins. And there are repeating units in chromatin called nucleosomes, which are made up of 146 base pairs of double helical DNA that is wrapped around a core of 8 histones. And there are 4 different types of histones within this structure of 8 that you should be aware of. And they're named H2A, H2B, H3, and H4. That's just the nomenclature they've been given. Now, acetylation occurs at the amino terminal tails of these histone proteins by an enzyme called histone acetyltransferase, which I'll just abbreviate as HAT. And this is a reversible modification. So the acetylation of histones is sort of kept in balance by another enzyme that removes these acetyl groups, which is called histone deacetylase, or HDAC-HDAC. The acetylation of histones leads to uncoiling of this chromatin structure, and this allows it to be accessed by transcriptional machinery for the expression of genes. On the flip side of this, histone deacetylation leads to a condensed or closed structure of the chromatin and less transcription of those genes. When these modifications that regulate gene expression are inheritable, they are referred to as the acetylation of histones leads to uncoiling of this chromatin structure, and this allows it to be accessed by transcriptional machinery for the expression of genes. On the flip side of this, histone deacetylation leads to a condensed or closed structure of the chromatin and less transcription of those genes. When these modifications that regulate gene expression are inheritable, they are referred to as epigenetic regulation. So when it comes to gene expression in DNA, you can basically think of DNA as coming in 2 flavors: densely packed and transcriptionally inactive DNA, which is called heterochromatin, and then less dense transcriptionally active DNA, which is euchromatin. And I like to think of heterochromatin as being densely packed and hibernating, like heterochromatin and hibernating both begin with H, kind of like a bunch of densely packed bears that are closed off in their cave for the winter, whereas euchromatin is waiting there with open arms, welcoming the transcriptional machinery to transcribe away. Now, often you will see that histone deacetylation is combined with another type of DNA regulatory mechanism, and that is DNA methylation. And this occurs in a process called gene silencing. And this is a more permanent method of sort of downregulating the transcription of genes. And DNA methylation involves the addition of a methyl group, which is a carbon with 3 hydrogens, to the cytosine DNA nucleotides by an enzyme appropriately called methyltransferase. And this typically occurs in cytosine-rich sequences called CPG islands. Don't forget that cytosine pairs with G, guanine, so that's why there are CG islands that you'll find. DNA methylation stably alters the expression of genes, and so it occurs as cells divide and differentiate from embryonic stem cells into specific tissues. And so this is essential for normal development and is associated with other processes such as genomic imprinting and X chromosome inactivation, topics for another discussion. And abnormal DNA methylation has been implicated in carcinogenesis, or the development of cancer. So you can see how the normal functioning of DNA methylation is a critical regulatory mechanism for our cells. Now, DNA methylation may affect the transcription of genes in 2 ways. First, the methylation of DNA itself may physically impede the binding of transcriptional proteins to the gene. And second, and likely more important, methylated DNA may be bound by proteins known as methyl CpG binding domain proteins, or MBDs for short. Now, MBD proteins can then recruit additional proteins to the locus or particular location in a chromosome, certain genes, such as histone deacetylases and other chromatin remodeling proteins. And this modifies the histones, forming condensed inactive heterochromatin that is basically transcriptionally silent. So what differences can't be attributed to that fact alone? The answer actually lies in the expression of that DNA, which genes are actively transcribed and which ones aren't. And there are several ways in which gene regulation occurs at the level of transcription, and so we're going to be talking about the main ones here. Now, let's draw out a hypothetical gene here, and associated with this gene is a sequence upstream, so towards the 3' region of the antisense strand, also called the template strand. And this sequence is called the promoter. And there's another sequence in between the promoter and the gene called the operator. The operator is the sequence of DNA to which a transcription factor protein can bind, and the promoter is the sequence of DNA to which the RNA polymerase binds to start transcription. Now, first off, in prokaryotes, we have what are called general transcription factors, which are a class of proteins that bind to specific sites on DNA to activate transcription. General transcription factors plus RNA polymerase and another protein complex called the mediator multiple protein complex constitute the basic transcriptional apparatus, which positions RNA polymerase right at the start of a protein coding sequence or a gene, and then releases the polymerase to transcribe the messenger RNA from that DNA template. Now, there's another type of DNA binding protein called activators, and these enhance the interaction between RNA polymerase and a particular promoter, encouraging the expression of the gene. And activators can do this by increasing the attraction of RNA polymerase for the promoter through interactions with subunits of the RNA polymerase, or indirectly by changing the structure of the DNA. An example of an activator is the catabolite activator protein, or CAP, and this protein activates transcription of the lac operon in E. coli. In the case of the lac operon in E. coli, cyclic adenosine monophosphate, or CAMP, is produced during glucose starvation. And so this CAMP actually binds to the catabolite activator protein, or CAP, which causes a conformational change that allows the CAP protein to bind to a DNA site located adjacent to the promoter. And then this activator, the CAP, then makes a direct protein-to-protein interaction with RNA polymerase that recruits the RNA polymerase to the promoter. Now, enhancers are sites on the DNA that are bound to by activators in order to loop the DNA in a certain way that brings a specific promoter to the initiation complex. And as the name implies, this enhances transcription of the genes in a particular gene cluster. And while enhancers are usually what are called cis-acting, cis meaning the same or acting on the same chromosome, an enhancer doesn't necessarily need to be particularly close to the gene that it acts on. And sometimes it's not even located on the same chromosome. Enhancers don't act on the promoter region itself, but are actually bound by activator proteins. And these activator proteins can interact with that mediator complex I mentioned earlier, which recruits RNA polymerase and the general transcription factors, which then can lead to transcription of the genes. So here I've drawn a little schematic of what it might look like to have the enhancer actually change the structure of the DNA so that the DNA is now looping around. Here you still have your promoter sequence, the operator sequence, the gene sequence, and the enhancer sequence, and having the DNA looped in such a way so that you then recruit RNA polymerase, the transcription factors, the mediator protein complex, and then you have transcription begin of this gene here. Now let's talk about repressors. Repressors are proteins that bind to the operator, impeding RNA polymerase's progress along the strand and thus impeding the expression of the gene. Now, if an inducer, which is a molecule that initiates gene expression, is present, then it can actually interact with the repressor protein in such a way that causes it to detach from the operator. And then this frees up RNA polymerase to then transcribe the gene further down on the DNA strand. One example of a repressor protein is the repressor protein associated, again, with the lac operon operator, which prevents the transcription of genes used in lactose metabolism unless lactose, which is the inducer molecule, is present as an alternative energy source. Now, silencers are regions of DNA that are bound by repressor proteins in order to silence gene expression. And this mechanism is very similar to that of the enhancer sequences that I just talked about. And similarly, silencers can be located several bases upstream or downstream from the actual promoter of the gene. And when a repressor protein binds to the silencer region of the DNA, RNA polymerase is prevented from binding to the promoter region. Now, a few notes about the differences between prokaryotes and eukaryotes when it comes to transcriptional regulation. And
a bit too much noise. Too much noise, man.
Prokaryotes, the regulation of transcription is really needed for the cell to be able to quickly adapt to the ever-changing outer environment that it is sitting in. The presence, the quantity, the type of nutrients actually determines which genes are expressed. And in order to do that, genes must be regulated in some sort of fashion. So a combination of activators, repressors, and rarely enhancers, at least in the case of prokaryotes, determines whether a gene is transcribed. In eukaryotes, transcriptional regulation tends to involve a combination of interactions between several transcription factors, which allows for a more sophisticated response to multiple conditions in the environment. And another major difference between eukaryotes and prokaryotes is the fact that eukaryotes have a nuclear envelope, which prevents the simultaneous transcription and translation of a particular gene, and this adds an extra spatial and temporal control of gene expression. Let's talk about post-transcriptional regulation, which is regulation basically once DNA has been transcribed into mRNA. And I've drawn out this little schematic for you here, and it kind of just shows you how a DNA strand has a corresponding RNA strand and then the mRNA strand afterwards. And I'll sort of explain what all the different colors and words mean in just a little bit. So once DNA is transcribed by RNA polymerase into the corresponding RNA strand, this RNA strand needs to get what I call a tidy little haircut and then don some protective outerwear before it can leave the comfort of the nucleus for its big debut into the cytoplasm in the form of a fully processed messenger RNA or mRNA strand. Now, keep in mind that this form of regulation occurs in eukaryotes only, and this modification also helps to stabilize the mRNA to protect it from premature degradation before it gets translated into a protein. So as you can see here, DNA gets transcribed one-to-one, base for base, into RNA. And you can see here that there are sections of the RNA that ultimately make it into the finished mRNA, these short segments which are termed exons. And they are the sequences that code for the ultimate protein product. And then there are short non-coding segments of RNA that get cut or spliced out. And this would be the haircut that I alluded to earlier. And so these are called introns.
Twice today
and this is accomplished by a large molecular entity called the spliceosome. So the spliceosome binds on either side of an intron, loops the intron into a circle, and then cleaves it off, and then ligates the two cut ends of the exposing exons together, kind of cinches them together. And an easy way to remember which sequences are exons, which ones are introns, is that exons exit the nucleus and introns stay in the nucleus. So exons kind of stands for exit. Now, even though the mRNA has gotten its nice little haircut, it's not quite ready to leave the nucleus just yet. It has to grab what is called a 5' cap and a 3' poly A tail. Now, what are those things that I just mentioned? So a 5' cap refers to changes at the 5' end of the mRNA. And remember that this is the phosphate end of the nucleotide bases in the mRNA. And some people like to remember this as F for 5' and for phosphate. So that's how you can kind of keep the two ends straight. And capping at the 5' end converts this end of the mRNA to a 3' end by a 5' to 5' linkage, which basically just protects the mRNA from exonucleases, which degrade foreign RNA. The cap also promotes ribosomal binding for translation and also helps the regulation of nuclear export of the mRNA. Now, the poly A tail, that goes on the other end, the 3' end of the mRNA, which has the terminal hydroxyl group. And so what do I mean when I say poly A tail? Well, poly A tail refers to polyadenylation, in which multiple adenosine monophosphates, or basically adenine bases, are added to act as a buffer for exonucleases in order to increase the half-life of mRNA and, again, protect it from degradation. And so the purpose of the poly A tail is really very similar to the 5' cap, which is basically to protect from degradation, help with promoting translation, and regulating nuclear export. The poly A tail also does one more thing, and it kind of just helps with transcription termination for the RNA polymerase that's transcribing the messenger RNA. Polyadenylation is catalyzed by an enzyme called polyadenylate polymerase, which adds the adenosine monophosphates using adenosine triphosphate as the substrate. And the poly A tail is built until it's about 250 or so nucleotides long. So overall, the 5' cap and the poly A tail help to stabilize the mRNA for translation. That's the key point to take home from here. So once the mRNA has donned its cap and tail and had its introns spliced out, it's now ready to exit the nucleus to be translated into a protein. Now, additionally, there is one more type of RNA regulation called RNA editing, which is a process that results in sequence variation in the RNA molecule and is catalyzed by various enzymes. RNA editing is relatively rare, and these events may include insertion, deletion, and base substitution of nucleotides within the edited RNA molecule. Now, one of these enzymes is called adenosine deaminase acting on RNA, or ADAR enzymes, which convert specific adenosine residues to inosine in an mRNA molecule by hydrolytic deamination. Another type of editing is called cytidine deaminase acting on RNA, or CDAR, which involves deamination of cytosine to uridine by cytidine deaminase. RNA editing is currently being extensively studied in relation to infectious diseases because the editing process alters viral enzymes and their function. So kind of an exciting new emerging concept in post-transcriptional regulation.
Perfect.
What is a non-coding RNA? Well, a non-coding RNA, or an mcRNA, as it is abbreviated, is a functional RNA molecule that actually skips this last step and is not translated into a protein. In other words, they just go directly from transcription into an RNA molecule and then go off to perform any number of vital functions within the cell. And there are many examples of non-coding RNAs, including microRNAs, ribosomal RNA, transfer RNA, the list goes on and on. And as we go through each of these different types and examples of non-coding RNAs, you'll start to see that there's sort of an emerging theme here. And that is that most of these non-coding RNAs participate in either transcription or translation in one capacity or another. So let's start off with microRNAs. MicroRNAs, or miRNAs, function in transcriptional and post-transcriptional regulation of gene expression. And they do this by base pairing with complementary sequences within mRNA or messenger RNA molecules. And this usually results in gene silencing through translational repression or target degradation. In essence, the mRNA to which these microRNAs bind are prevented from being translated, or they are sent on a pathway for degradation. Now, the next set of non-coding RNAs that we'll be talking about are all involved in translation. The first of which is ribosomal RNA. Now, ribosomes are the cellular machinery used to translate mRNA into proteins, and it is made up of one type of RNA molecule, ribosomal RNA. Transfer RNAs are an adapter molecule that links the codons in an mRNA strand to the corresponding amino acids. And so this is another type of non-coding RNA that you'll see in translation. The third type is called SNO RNA, which stands for small RNA molecule ribosomal RNA. Transfer RNAs are an adapter molecule that links the codons in an mRNA strand to the corresponding amino acids. And so this is another type of non-coding RNA that you'll see in molecule ribosomal RNA. Transfer RNAs are an adapter molecule that links the codons in an mRNA strand to the corresponding amino acids. And so this is another type of non-coding RNA that you'll see in translation. The third type is called SNO RNA, which stands for small nucleolar RNA. And it's a class of small RNA molecules that guide covalent modifications of ribosomal RNA, transfer RNA, and small nuclear RNAs, primarily through methylation, which is the addition of methyl groups, or pseudouridylation, which is the addition of an isomer of the nucleoside uridine. Now, another class of non-coding RNAs are the small nuclear RNAs, or snRNAs, not to be confused with the SNO RNAs, the small nucleolar RNAs that we just talked about. Now, small nuclear RNAs get their name from the fact that the average length of these RNA molecules is approximately 150 nucleotides. And their primary function is in the processing of pre-mRNA in the nucleus. They also aid in the regulation of transcription factors or a particular RNA polymerase 2, as well as maintaining telomeres, which are the regions of repetitive nucleotide sequences at the end of a chromatid, which protects the end of the chromosome from deterioration during chromosomal replication. Now, small nuclear RNAs get their name from the fact that the average length of these RNA molecules is approximately 150 nucleotides. And their primary function is in the processing of pre-mRNA in the nucleus. They also aid in the regulation of transcription factors or a particular RNA polymerase 2, as well as maintaining telomeres, which are the regions of repetitive nucleotide sequences at the end of a chromatid, which protects the end of the chromosome from deterioration during chromosomal replication. Now, snRNA can be associated with a set of specific proteins and form complexes that are called small nuclear ribonuclear proteins, or snRNPs, or sometimes people just call them SNRPs. And there is a special SNRP complex called the spliceosome, made up of 5 small nuclear RNAs and over 150 proteins, that is responsible for splicing or removing the introns contained in messenger RNA, which is a major step in the post-transcriptional modification that takes place in the nucleus of eukaryotes. And the way that the spliceosome does this is that it binds to specific sequences in the pre-messenger RNA strand and performs 2 sequential trans-esterification reactions that splice out the intron and then ligate the 2 exons to form a mature mRNA. So now you know a little bit more of some examples of non-coding RNAs and some of the functions that they perform within the cell. Oncogenes are genes that code for proteins that normally direct cell growth. They start out as proto-oncogenes, and then something happens to convert that proto-oncogene into a full-blown oncogene. In essence, some sort of tumor-inducing agent, or it could also be totally spontaneous. Now, proto-oncogenes code for proteins that help to regulate cell growth and differentiation, which makes sense since the essence of a tumor, whether it is a cancerous or benign one, is unregulated cell growth. In fact, the words oncogene and oncology, which is the study of cancer, share the same root word, onkos, which is Greek for mass or bulk. Now, the products of these genes are often involved in signal transduction and the execution of mitogenic signals. And you should recall that a mitogen is a chemical substance that encourages a cell to start cell division, basically something that triggers mitosis. So how does a proto-oncogene make that switch into an oncogene? Well, there are 3 main mechanisms: deletion or point mutation, gene amplification or increased mRNA stability, and chromosomal rearrangement. So let's start with the first one. Deletion or point mutation encoding sequence of the DNA in the gene itself or within a regulatory region, such as a promoter region, can lead to either a protein that is produced in the same normal amounts but is hyperactive for some reason, or maybe there is some sort of loss of regulation and the normal protein is just overexpressed. Gene amplification, or an increase in mRNA stability that prolongs the existence of the mRNA and thus its activity in the cell, can lead to a normal protein that is overexpressed. And then finally, chromosomal rearrangement involves translocation of a gene to a nearby regulatory sequence that then causes this normal protein, the gene product, to be overexpressed. Or you could possibly have fusion to an actively transcribed gene, which overexpresses the fusion protein or leads to a hyperactive fusion protein. And so really, you can start to see a theme here emerging where the key idea is that you have either a normal protein that is just overexpressed, basically too much of the normal protein, or you can have normal expression, but the protein itself is just a hyperactive one. Now, there are tons of examples of oncogenes, including the SARC oncogene, RAS, MYC, receptor tyrosine kinase, and also cytoplasmic tyrosine kinases. So let's talk about each one of those here. SARC was the first confirmed oncogene, which was discovered in 1970 and was termed SARC for sarcoma, which is a tumor of mesenchymal cells or connective tissue cells. It was actually an oncogene discovered in a chicken retrovirus. And so SARC codes for a non-receptor protein tyrosine kinase, so it phosphorylates specific tyrosine residues in other proteins. Another example of an oncogene is the RAS oncogene, which codes for a small GTPase, which hydrolyzes GTP into GDP and phosphate. This protein is activated by growth factor signaling and functions like a binary switch, sort of an on-off switch in growth signaling pathways. Examples of downstream effectors of RAS includes the protein MAPK, which is a type of kinase that in turn regulates genes that mediate cell proliferation.
Yes,
You can see examples of RAS oncogene mutations in thyroid tumors, certain leukemias, and cancers of the pancreas and colon. The MYC oncogene codes for a transcription factor that induces cell proliferation. And there's a very common translocation involving MYC between chromosomes 8, where the MYC oncogene is found, and 14, which leads to a certain type of lymphoma called Burkitt's lymphoma. The RTK oncogene stands for receptor tyrosine kinase, which is responsible for adding phosphate groups to other proteins to turn them on or off. These are similar to the SRC gene, which is a non-receptor tyrosine kinase, the difference being the location of the protein, either at the cell surface as a receptor or not as a receptor. So say you have a cell surface receptor that receives a signal from outside of the cell. Well, then that signal gets propagated or transmitted into the cell via these receptor tyrosine kinases that add phosphate groups to the target protein, specifically on tyrosine residues. And they can cause cancer by turning a receptor constitutively or permanently on in the absence of signals from outside the cell. Some well-known examples of receptor tyrosine kinases are vascular endothelial growth factor, or VEGF, epidermal growth factor, or EGFR, and platelet-derived growth factor, PDGF. And finally, we have the cytoplasmic tyrosine kinases, which mediate responses to the activation of receptors of cell proliferation, migration, differentiation, and survival. A well-known example is the BCR-ABL gene in chronic myelogenous leukemia, or CML, also known as the Philadelphia chromosome. It is a fusion of parts of DNA from chromosome 22 and chromosome 9. So basically, a part of chromosome 22, which contains the BCR gene, fuses with a fragment from chromosome 9, which contains the ABL gene. When these two chromosome fragments fuse, the genes also fuse, creating this new gene, the BCR-ABL gene. The fused gene codes for a protein that displays high protein tyrosine kinase activity, which is actually due to the ABL half of the protein. And the unregulated expression of this protein activates other proteins that are involved in cell cycle and cell division, which causes a cell to grow and divide uncontrollably, basically becoming cancerous. So as a result, the Philadelphia chromosome is associated with chronic myelogenous leukemia, a certain type of leukemia, as I mentioned before, as well as other forms of leukemias, which are cancers of white blood cells.
I think let's do this one, and then we might watch. I think I want to rewatch DNA and DNA replication. I think I'm okay with the rest, and then we'll move on. We'll do gene mutations. We'll do Mendelian. We'll do meiosis, and we'll do DNA tech. Is that it? That's it. I mean, that feels like. It feels like. Is it 4? Mutations, genetics. Oh, and we have to do chromosomal inheritance. No, and we have to do chromosome. Which one? Evolution and population dynamics. No, chromosomal inheritance. Where's chromosomal inheritance? Oh, I think it's this one. Oh, okay. It's kind of switched. Wait. Mendel genetics. And then we skip. Oh, DNA tech, and then, okay. 1, 2, 3 after DNA tech. Oh my god, I'm gonna write this down. I can't. It needs to be. Okay, we have to do today. Oh, let's try it here actually. Genetic mutations. Mendelian genetics. DNA tech. And chromosomal inheritance. Let's do those 4 today. Today, Junior. All right, genetic mutations, Mendel genetics. Oh, and then 11 and then 12. 11 and then 10. 11 and then 10. Whereas this is 9 and then this is 8. great. They're great. Tumor suppressors. Let's refresh. And watch the final video for this one.
Tumor suppressor genes are those genes whose protein products either have a halting effect on the regulation of the cell cycle, or they can also promote apoptosis, or sometimes both. So in other words, these proteins are like big stop signs that act as safety checks to help stop the mistakes in cell division that could lead to uncontrolled cell growth in cancer. Now, there are several sort of categories of tumor suppressor proteins. Those that recognize DNA damage and either repair it or initiate programmed cell death, apoptosis, if it can't be repaired, so DNA repair proteins. Then there are those proteins that act as repressors of genes that are essential for the continuation of the cell cycle. So if these genes are actively repressed and thus not expressed, the cell cycle does not continue on. So you have cell cycle repressors. With tumor suppressors, there is this concept called the two-hit hypothesis, in which both alleles, and remember that alleles are basically the copies for a certain gene. You have two copies for any given gene, one on the chromosome that you got from your mom and one on the chromosome that you got from your dad. Now, in the two-hit hypothesis, both alleles must be mutated before the effect is manifested. Because if only one of the alleles for the gene is damaged, then you have this sort of backup second copy that can still produce the protective protein. So you need two hits, one hit for each of the alleles that you have. Another way you can think of this is that in mutated oncogenes, these alleles are typically dominant. So a mutation in only one of the alleles yields the cancerous phenotype. But with a mutated tumor suppressor allele, these mutations are recessive because both alleles must be mutated in order to lead to the cancerous phenotype. The two-hit hypothesis was first proposed with cases of retinoblastoma, a rapidly developing cancer that originates from the immature cells of the retina, the light-detecting tissue of your eye. And I'll write this as PRB for retinoblastoma protein. Now, the retinoblastoma protein prevents the cell from replicating when its DNA is damaged. And it does this by preventing progression of the cell cycle from G1 into the S phase, or synthesis phase. So the retinoblastoma protein binds and inhibits transcription factors, which normally push the cell into the S phase. And this complex acts as a growth suppressor, and so the cell remains in the G1 phase. This complex also attracts a histone deacetylase protein to the chromatin, which reduces transcription of S phase promoting factors. And you can remember this by recalling that histone deacetylase leads to chromatin condensation, or transcriptionally inactive, the S phase. There's a growth suppressor, and so the cell remains in the G1 phase. This complex also attracts a histone deacetylase protein.
Histone deacetylase, which means it's going to deacetylate.
Into the chromatin, which reduces
HDAC. I think it was what was it? Let me see real quick. Oh yeah, so acetyl group will loosen it, deacetylase will tighten it, HDAC, heterochromatin.
transcription of S phase promoting factors. And you can remember this by recalling that histone deacetylase leads to chromatin condensation, or transcriptionally inactive chromatin. So this also further suppresses DNA synthesis.
Okay.
Another very well-known tumor suppressor protein is the P53 protein. Homozygous loss of this protein is found in up to 65% of colon cancers, 50% of lung cancers, and also in breast cancer. So this is clearly a very critical tumor suppressor protein. And so P53 activates DNA repair proteins when DNA has sustained damage. And it can also arrest growth by holding the cell cycle hostage, if you will, at the G1 to S regulation point. And this gives DNA repair proteins some time to fix the damage and allow for continuation of the cell cycle. So specifically, P53 binds DNA and activates several genes, including ones that code for a protein called P21, which binds the cyclin CDK, or cyclin-dependent kinase complex, which is actually the complex responsible for pushing the cell from the G1 to S phase in the cell cycle. P53 also functions in the initiation of apoptosis if the damage to DNA is irreparable. One significant exception to the two-hit rule for tumor suppressor genes is with certain mutations of the P53 gene product, which can then result in what is called a dominant negative, meaning that a mutated P53 protein can prevent the protein product of the normal allele from functioning. So don't forget to sort of keep that in the back of your mind when you're thinking about tumor suppressor genes.
Okay, thank you, Lee. Oh, genetic mutation. What is this one? No, I like the guide summary. Right, guide summary. go here. I want to watch that. I want to just rewatch quickly this one. Maybe just from up to here with Sal, I guess. Well, I don't know if we have to rewatch.
Something that stores information always pairs with the base timing. Pair. Humans, 3 pairs. And then you would have this a protein. If you digested what a double helix looks like, this is it.
Sure, whatever.
We already have an overview video of DNA, and I encourage you to watch that first. What I want to do in this video is dig a little bit deeper, actually get into the molecular structure of DNA. And just as a starting point, let's just remind ourselves what DNA stands for. I'm going to write the different parts of the word in the different colors. So it stands for deoxyribonucleic, as one of the should say the backbones of, or one side of the ladder. This is the other side of the ladder, and then each of these bridges, and we'll talk about what there are just here. So this can here, this double bond is also word, and so that's what it means. A charge because its electrons are being.
Fine. Let's check this one out.
Then you have options to potentially sop up already deprotonated. But in general, phosphate groups are considered, there's steam here, this is actually an actual thing, or the reason why but in general, especially see that when you hide the different directions. So the carbon, but you see, so 1', 2', 3, just the fascinating molecule. We could speak for days about it. It's actually mind-blowing when you think about its implications for who we are. Let's talk a little bit in more depth about how DNA actually copies itself, how it actually replicates. And we're going to talk about the actual actors in the process. Now, as I talk about it, I'm going to talk a lot about the 3' and the 5' ends of a DNA molecule. And if that is completely unfamiliar to you, I encourage you to watch the video on the anti-parallel structure of DNA. And I'll give a little bit of a quick review here, just in case you saw it, but it was a little while ago. This is a zoom in of DNA. It's actually the zoom in from that video. And when we talk about the 5' and 3' ends, we're referring to what's happening on the riboses that form part of this phosphate sugar backbone. So we have ribose right over here, 5 carbon sugar, and we can number the carbons. This is the 1' carbon, that's the 2' carbon, that's the 3' carbon, that's the 4' carbon, and that's the 5' carbon. So this side of the ladder, you could say, let me draw a little line here. This is going in the 3' to 5' direction. So this end is 3', and then this end is 5'. So 3' to 5'. Notice this phosphate connects to the 3', then we go to 5', connects to a phosphate. This connects to a 3', and it connects, and then we go to the 5', connects to a phosphate. Now on this end, as we said, it's anti-parallel. It's parallel, but it's oriented the other way. So this is the 3'. This is the 5'. This is the 3'. This is the 5'. And so this is just what we're talking about when we talk about the anti-parallel structure. These two backbones, these two strands are parallel to each other, but they're oriented in opposite directions. So this is the 3' end, and this is the 5' end. And this is going to be really important for understanding replication, because the DNA polymerase, the things that's adding more and more nucleotides to grow a DNA strand, it can only add nucleotides on the 3' end. So if we were talking about this right over here, we would only be able to add going that way. We wouldn't be able to add going that way. So one way to think about it is you can only add nucleotides on the 3' end, or you can only extend DNA going from 5' to 3'. If you're only adding on the 3' end, then you're going from the 5' to the 3' direction. You can't go from the 3' to the 5' direction. You can't continue to add on the 5' side using polymerase. So what am I talking about with polymerase? Well, let's look at this diagram right over here that really gives us an overview of all of the different actors. So here is just our DNA strand, and you can imagine it's just somewhat natural, in its natural unreplicated form. And you can see we've labeled here the 3' and the 5' ends, and you could follow one of these backbones. This 3', if you follow it all the way over here, it goes, this is the corresponding 5' end. So this and this are the same strand, and this one, if you follow it along, if you go all the way over here, is the same strand. So this is the 3' end and 3' end of it, and then this is the 5' end of it. Now, the first thing, we've talked about this in previous videos where we give an overview of replication, is the general idea is that the two sides of our helix, the two DNA, the double helix needs to get split, and then we can build another side of the ladder on each of those two split ends. You could really view this as, if this is a zipper, you unzip it, and then you put new zippers on either end. But there's a lot of, in reality, it is far more complex than just saying, oh, let's open a zipper and put new zippers on it. It involves a whole. In the earlier video on DNA replication, we go indeed, and this can only go for brains. These aren't computers. They don't know you're amazing. What you plus base pair, you want, okay, this is the cells take. So this would still produce a lot of errors, but then the error to better in all of the cells, or most of the cells.
Here is a pair of chromosomes as they would appear during mitosis, and the ends of chromosomes are capped with an area. Here's a pair of chromosomes as they would appear during mitosis, and the ends of chromosomes are capped with an area known as telomeres. And telomeres are mainly found in eukaryotic chromosomes because usually prokaryotes just have one circular chromosome, so it doesn't have any ends. And what do telomeres do? Well, they protect chromosomes, or protect the ends of chromosomes from deterioration. Why would the ends of chromosomes deteriorate? So the enzymes that replicate chromosomes are not able to get to the very, very end of the chromosome. So they're going to get to, let's say, this area. So there's going to be a small spot over here that's not replicated. And since the telomeres don't have any genes in them, that's not really harmful. It doesn't really matter. So what would happen if there were no telomeres? Well, let's take a look at the other chromosome. If there were no telomeres, and let's say the chromosome was only replicated till about here, there would be this area with useful genes that wouldn't be replicated, and that would be pretty problematic. So basically, telomeres act as a buffer zone because they do not contain any important genes. Another thing that telomeres do is they prevent chromosomes from sticking to each other. If chromosomes stuck to each other, then a lot of the genes would be scrambled and genes wouldn't be where they're supposed to be, and that would be pretty problematic. And here's actually a picture of human chromosomes where the telomeres are highlighted in this fluorescence. So you can see the telomeres over here. So you can see at both ends of each chromatid, there are telomeres. And so what happens is that with each time the chromosomes replicate, the telomeres get a little bit shorter and shorter and shorter. So there's an enzyme known as telomerase. And telomerase is able to lengthen telomeres and bring them back to their original length. So there are some cells that replicate a lot and they have a lot of telomerase, so the cell can keep on replicating and replicating. But then there are other cells that do not have a lot of telomerase, and when the telomeres are basically non-existent anymore because the chromosomes replicated many, many times, a lot, and they have a lot of telomerase, so the cell, what happens is that with each time the chromosomes replicate, the telomeres get a little bit shorter and shorter and shorter. So there's an enzyme known as telomerase. And telomerase is able to lengthen telomeres and bring them back to their original length. So there are some cells that replicate a lot and they have a lot of telomerase, so the cell can keep on replicating and replicating. But then there are other cells that do not have a lot of telomerase, and when the telomeres are basically non-existent anymore because the chromosomes replicated many, many times, let's just get rid of the telomeres. So the chromosomes will actually not be able to replicate, and so the cell will not divide again, and it will kind of die. Now that we're talking about telomeres, I want to bring up a topic that's tangentially related, and that is single copy DNA and repetitive DNA. So single copy DNA is when you have a DNA sequence. I'm just going to make one up. Let's say A, T, C, C. That basically does not repeat itself, so it might be flanked by other DNA sequences, as opposed to repetitive DNA, which is when you have a DNA sequence that keeps repeating itself. So you might have an ATCC, and then again, ATCC, ATCC, etc. So what's the difference between single copy DNA and repetitive DNA? So here we have a spectrum. On the left, we have single copy DNA. In the middle, we have DNA that's somewhat repetitive. And on the right, we have highly repetitive DNA. So single copy DNA holds most of the organisms, there's an apostrophe there, important genetic information. So basically, most of the important genes are going to be single copy. So since the important genes are single copy DNA is transcribed and translated, and it has a low mutation rate, which is a good thing because, of course, we don't want there to be mutations in the important genes. Repetitive DNA, or DNA that's somewhat repetitive, is found, well, at least in mammals and in insects, near the centromeres. If you recall, the centromeres are the center of the chromatid, or when you have chromatids that are duplicated, the chromatids are attached by the centromere, by that middle part in the chromosome. And they may contain genes that are transcribed and translated, but then there might also be parts of the repetitive DNA that don't contain genes, and those parts are not transcribed and translated. And repetitive DNA has a higher mutation rate than single copy DNA. Now let's take a look at DNA that's highly repetitive. So it contains no genes, and because it contains no genes, it is not transcribed and not translated. And highly repetitive DNA has an even higher rate of mutation than DNA that's somewhat repetitive. So there's lots of highly repetitive DNA that we're not exactly sure what its purpose is. Scientists are currently trying to figure out what the purpose of this highly repetitive DNA is, but there are some sections of highly repetitive DNA that we do know what their purpose is. For example, telomeres. Telomeres are sections of highly repetitive DNA, and as we explained before, their purpose is to basically act as a buffer zone for the important part of the chromosome. And in fact, the DNA sequence that's repeated in telomeres is this right over here, GGTTAG. And in human chromosomes, the telomeres are made up of approximately 2,000 repeats of this DNA sequence, GGTTAG.
Okay, that was fast.
Controllably.
2.
Let's take a look at a segment of DNA that's in the process of being replicated. And I want to focus in particular on the enzyme that replicates DNA. And that enzyme is DNA polymerase. And actually, there are a few different types of DNA polymerases, and the one that we're looking at right now is DNA polymerase 3. So DNA polymerase 3 synthesizes new DNA, and it also has the ability to proofread, or kind of check the DNA that's putting together and make sure that there are no mistakes in it. But before we get into that, let's just orient ourselves and quickly summarize the diagram that we're looking at. So this enzyme over here is DNA helicase. That's the enzyme that unwinds the double-stranded DNA so that DNA polymerase can then come in and start replicating. Right over here, you can see I drew the backbone in a different color. That's the RNA primer. And let's just label the DNA strand that's being synthesized. It's synthesized from 5' to 3'. And actually, the bottom strand of DNA is synthesized in the same time as the top strand, but I just left that out of the drawing to keep things simple. And let's say that the yellow bases represent the nitrogen-based thymine. Let's say that the orange bases represent cytosine. The green ones represent adenine. And the blue ones represent guanine. And so thymine and cytosine are the pyrimidines. They are composed of a single ring structure. So they're made up of one ring that has 6 sides to it. And then adenine and guanine are the purines.
Yeah.
They are a double ring structure. So they're composed of one ring with 6 sides to it, and then that ring is attached to another ring that has 5 sides to it. And actually, these structures are a little bit more complex. There are other atoms, in it, and there are some double bonds, but we're just going to keep things simple for now and leave it at that. And so let's get back to our DNA that's being replicated. Right over here, I left a space. I didn't put the nucleotide in. And let's say that by accident, instead of it being paired up with the proper base, which is adenine, it accidentally gets paired up with a guanine. So that's a mistake. And DNA polymerase 3 actually has the ability to sense if it made a mistake. And if it does realize that, it's going to go backwards, and it's going to actually remove the incorrect base and replace it with the correct base. So let's do that. It's going to remove the incorrect base and replace it with the correct one. Of course, remember the nitrogen base is attached to the sugar backbone. And so this activity that I just described to you is called exonuclease activity. And so nuclease tells us that means the ability to remove a nucleotide. And exo, just going to underline that. So exo tells us that it can remove a nucleotide, but only from the end of a DNA strand. So it was able to remove the nucleotide because it was at the end of the strand. This is in contrast to endonuclease activity. So an endonuclease can actually remove a nucleotide from the middle of a DNA strand. So it would be able to remove, a nucleotide from like right over here, for example. And just keep that in mind because we're going to come across some endonucleases as well. Anyway, back to our exonuclease activity. If we want to be more specific, the exonuclease activity of DNA polymerase 3 is actually 3' to 5' exonuclease activity. And the reason it's called 3' to 5' exonuclease activity is because when DNA polymerase 3 makes that correction, it has to move backwards in the 3' to 5' direction in order to do that. There's another enzyme, DNA polymerase 1. I'm just going to abbreviate polymerase with POL. And DNA polymerase 1 also has exonuclease activity. And DNA polymerase 1 is actually the enzyme that will remove the RNA primer at the end of replication. And just as a side fact, the exonuclease activity of DNA polymerase 1 is actually in the 5' to 3' direction. So if you want, you can just keep that in mind. And so DNA polymerase 3 and DNA polymerase 1 are both able to repair or fix mistakes that happen during DNA replication. And just to give you some perspective as to how often this occurs with and without repairs. So normally we'll have a mistake happening in replication between 1 in 100,000 bases to 1 in 1 million bases. That's normally the amount of mistakes that would occur. But with the repair mechanisms of DNA polymerase 3 and DNA polymerase 1, this is reduced to a mistake that happens once in about 100 million bases. And so they are very, very effective at lowering the error rate in DNA replication. And so the next question I want to ask is, what if this mistake over here was somehow not corrected during replication? Maybe there was something wrong with one of the enzymes, something happened, and that mistake was actually sustained. So let's take a look at that. So here's a piece of DNA with our mistake incorporated into it. And so before we discuss if this mistake can be corrected or not, let's see what happens if this mistake is not corrected. So right here we have our original DNA, and we're replicating it. And let's just say that this strand over here is the same as that strand. Let's see that the bottom strand in our original DNA is the same as this strand. And so let's look first at the newly replicated DNA on the left. So we have right over here the thymine base, and assuming DNA was replicated properly, it's going to have an adenine complementary to it. Now let's take a look at the DNA on the right. So on the bottom, we had a guanine, and it's going to be paired up, hopefully with the correct base, which is a cytosine. Now let's just quickly look back at our original DNA. We were supposed to have a thymine with this complementary adenine. And actually, that's exactly what we got over here. Just gonna circle it. So this DNA is actually in the correct sequence. But look at the DNA over here on the right. This is not correct. This is a mutation. And this is an example of how mutations can occur if the DNA repair mechanisms are not working properly. And so let's go back to our original question. Can we fix the original mistake so that this mutation does not occur? And the answer to that question is yes. So fortunately, our cells have what's called the mismatch repair mechanism. And the mismatch repair mechanism is composed of a number of proteins. And the first thing these proteins are going to do is they're going to recognize if there's a problem. And the reason that they're able to recognize the problem is that when you have a mismatch in DNA, it tends to distort the sugar backbone a little bit. And they're going to mark the area with a cut. So they're going to cut the incorrect base or mark it with a cut. The next thing that's going to happen is an exonuclease is going to remove the incorrect nucleotide. So we're going to remove the incorrect nucleotide. The next step is one of the DNA polymerases is going to insert the correct nucleotide. So we're going to pair our thymine up with adenine. And the last step is a DNA ligase is going to connect the new nucleotide to the nucleotides on its sides and also to its complementary nucleotide on the other strand. And I'm actually going to just correct that distorted sugar backbone. And so here's our repaired DNA. And just to clarify, the mismatch repair mechanism that we're talking about here happens after replication. The repair is done by DNA polymerase 3 and DNA polymerase 1 that we discussed before. That happens during replication or at the end of replication. And so one thing you might be wondering is, how does the mismatch repair mechanism know to distinguish between the original parental strand and the newly synthesized strand that has the mistake on it?
Right.
In other words, how does it know which base over here is correct? In our case, that's the thymine, and which one is incorrect? In our case, well, it was guanine. And we know the answer to that question in bacteria. So in bacteria, the parental strand will have adenines that are methylated. So I'm just going to draw some methyl groups on all the adenines. And that allows the mismatch repair mechanism to kind of recognize and distinguish between the original strand that has the correct base on it and the new strand that has the incorrect base on it. Mm-hmm. But we're not quite sure how the mismatch repair mechanism in eukaryotic cells and in other prokaryotic cells knows to distinguish between the strand that has the correct nucleotide and the strand that has the incorrect nucleotide.
We're just not sure, huh? What the heck? What the heck?
There are certain things that can cause damage to the structure of DNA. And one example of that is UV rays. And so before we get into the damage that UV rays cause, let's just focus for a second on the key that I drew here on the left. And it's just to help us remember which colors represent which nitrogen bases. So yellow represents the nitrogen-based thymine, the orange bases represent cytosine, the green bases represent adenine, and the blue bases represent guanine. And so back to our DNA that's being damaged. If you look over here, there are two thymine bases that are kind of stuck together. And that's called a pyrimidine dimer. So a dimer is simply when you have two molecules that are identical, they're stuck to each other. And pyrimidine tells us that it can be two thymines that are stuck together, or it can be two cytosines that are stuck together. And UV rays cause the formation of pyrimidine dimers. And so you can see that the pyrimidine dimer actually is causing the sugar phosphate backbone to protrude or kind of stick outwards. And not just that, but because the backbone is sticking outwards, the bond between this cytosine and this guanine snapped. So you can see that there's some structural damage that happened to the DNA. And so what are some factors that can cause damage to DNA? And I want to just make a clear distinction between a mutation and DNA damage. So it's not the same thing. A mutation is when you have a change in the sequence of DNA. So for example, if we had a piece of DNA that read A, T, C, G, and then something happened and read A, C, G. So this A is in the wrong place. That is a mutation. But when we talk about DNA damage, we're talking about damage to the structure of DNA, but the nucleotides are actually in the correct order. And so DNA damage can be caused by endogenous or internal factors, and that means factors that originate within us, within our own cells. So for example, there are certain byproducts of metabolism that can cause DNA damage, or DNA damage can be caused by exogenous or external factors, and those are factors that originate outside of us or outside of the organism that we're discussing. So let's start with exogenous factors first. So we spoke about one of them, UV rays, and there are a lot of exogenous factors that cause DNA damage, but we're just going to list a few. Gamma rays can cause DNA damage, X-rays, and so that's why it's not healthy to be exposed to a lot of these rays. And now let's talk about some endogenous factors. So reactive oxygen species is an example of an internal factor that can cause DNA damage. And reactive oxygen species are molecules that contain oxygen, and they're highly, highly reactive. So there are a lot of different kinds of reactive oxygen species, but we're just going to give two examples. So for example, a superoxide anion, which is O2 with a negative charge. So let's just draw that. It's two oxygen atoms that are bound together, but there is one extra electron. And I'm actually going to draw the extra electron in a different shade of purple. And so this whole molecule has a negative charge. So that's a reactive oxygen species. Another example would be peroxides. So peroxides are molecules that have two oxygens, and on either end there's another atom. So that R can represent the different types of atoms. So this is the general way that a peroxide looks. You might have heard of hydrogen peroxide. So this is hydrogen peroxide. And so where are these reactive oxygen species in our cells coming from? So actually, reactive oxygen species are a normal byproduct of the electron transport chain in the mitochondria. So there are a lot of reactive oxygen species all over our cells. But fortunately, we have many enzymes that help protect against the damaging effects of reactive oxygen species. And you may have heard of the term
Whoa, O2 is a superoxide anion. What? Interesting.
antioxidant
Peroxide.
product of the electron transport chain in the mitochondria. So there are a lot of reactive oxygen species all over our cells. But fortunately, we have many enzymes that help protect against the damaging effects of reactive oxygen species. And you may have heard of the term antioxidant. And so an antioxidant is a molecule that also helps protect us against the damaging effects of reactive oxygen species. You may have heard that certain foods are really healthy because they have a lot of antioxidants, and that's true. So vitamin C, for example, is an antioxidant. Vitamin E, and there are many different types of antioxidants, but we're just going to give these two as an example. And so now that we've discussed some of these sources of DNA damage, let's go back to our damaged DNA and see if there's a way to fix this. So our cells can get rid of these pyrimidine dimers in a process called nucleotide excision repair. And so the first step in nucleotide excision repair is an enzyme, an endonuclease, is going to remove the pyrimidine dimers and any other nucleotides that are kind of not the way they're supposed to be. And so I just want to pause for a second and analyze that word. So nuclease tells us that it's an enzyme that's able to cut out nucleotides. And that prefix endo tells us that it's able to cut out nucleotides from within a DNA molecule. That many enzymes that help protect against the peroxides. So peroxides are molecules that have two oxygens, and on either end there's another atom. So that R can represent the different types of atoms. So this is the general way that a peroxide looks. You might have heard of hydrogen peroxide. So this is hydrogen peroxide. And so where are these reactive oxygen species in our cells coming from? So actually, reactive oxygen species are a normal byproduct of the electron transport chain in the mitochondria. So there are a lot of reactive oxygen species all over our cells. But fortunately, we have many enzymes that help protect against the damaging effects of reactive oxygen species. And you may have heard of the term antioxidant. And so an antioxidant is a molecule that also helps protect us against the damaging effects of reactive oxygen species. You may have heard that certain foods are really healthy because they have a lot of antioxidants, and that's true. So vitamin C, for example, is an antioxidant. Vitamin E, and there are many different types of antioxidants, but we're just going to give these two as an example. And so now that we've discussed some of these sources of DNA damage, let's go back to our damaged DNA and see if there's a way to fix this. So our cells can get rid of these pyrimidine dimers in a process called nucleotide excision repair.
Nucleotide excision repair. Okay.
And so the first step in nucleotide excision repair is an enzyme, an endonuclease, is going to remove the pyrimidine dimers and any other nucleotides that are kind of not the way they're supposed to be. And so I just want to pause for a second and analyze that word. So nuclease tells us that it's an enzyme that's able to cut out nucleotides, and that prefix endo tells us that it's able to cut out nucleotides from within a DNA molecule. That's in contrast to an exonuclease that can only take out nucleotides that are at the beginning or end of the DNA molecule.
Okay.
But anyway, the endonuclease is going to cut out the dimer and any other nucleotides that are not properly arranged. So let's just cut out all these nucleotides. The next step is a DNA polymerase. I'm just going to abbreviate that, POL, is going to come and bring the nucleotides that belong there. And then the last step is DNA ligase, is going to make sure that those new nucleotides are attached properly to the nucleotides on either side and also to the nucleotide that's complementary on the other strand. And so that was a mouthful, but let's actually just draw all of that. So let's get rid of our backbone that's kind of protruding and just not right. So let's redraw our backbone, something like that, or actually put a little bit closer. And then DNA polymerase brings the correct nucleotides. But remember, it's the ligase that actually connects the nucleotides properly. And so here's our corrected DNA. But what happens if for some reason the nucleotide excision repair is not working properly? And this repair mechanism is only one example. There are many different types of DNA damage that can occur and many different types of repair mechanisms. What happens if for some reason one of these or a couple of these are not working properly? then we get a cell that has a lot of damaged DNA. And there are three things that can happen to a cell like this. The first is it might go into this dormant state where it just ages and does not divide anymore. That's called senescence. The second thing that might happen to it is what's called programmed cell death, or apoptosis. And that basically means that the cell is going to commit suicide and die. And the third thing that might happen is the cell might start to divide uncontrollably. I'm going to write unregulated cell division. And this can cause cancer. And so actually the skin cancer melanoma is an example of this. Melanoma, that's an N. Melanoma happens when the nucleotide excision repair mechanism that we just discussed is not working properly. And so you have this accumulation of pyrimidine dimers that damages the DNA very much, and then the cell starts to divide uncontrollably.
Okay.
Let's take a piece of DNA, and let's just unwind it into its two strands, and just help us remember that DNA is really a very, very long molecule. I'm going to put arrows here on our two strands of DNA. And the question I want to ask you is, if we were to replicate this DNA, what would the end result look like? So I'm kind of skipping over the entire process of how the DNA is replicated and focusing just on the product. And so we have three choices. The first is conservative replication. And in conservative replication, we have our old pair of DNA, and then we synthesize a completely new pair of DNA. So you can see the old pair that looks just the same as what we had before in yellow, and then we have a completely new pair, which is represented in blue. Our next choice is dispersive replication. And in dispersive replication, we're going to end up with two pairs of DNA. And in each one of those pairs, we have some old DNA and new DNA dispersed within that double-stranded DNA. So you can see there's yellow and blue mixed up together. And it wouldn't necessarily have to be in the ratio that I drew it in. I drew it in this kind of neat ratio where the yellow and blues are the same size, but perhaps the yellows would be a little bit bigger and maybe some of the blue parts smaller, or vice versa. And the third option we have is semi-conservative replication. And in semi-conservative replication, each pair has one old strand that you see in yellow, of course, and one new strand that's in blue. And this question was answered by two scientists, one by the name of Meselson and one by the name of Stahl. And they conducted a famous experiment which was named after them. So the Meselson-Stahl experiment. And in this experiment, they proved that DNA replication is semi-conservative. So this is how DNA is replicated.
Perfect. Short and simple. Let's go ahead and reread this one, and then we'll watch the videos. I think when I read it, I was kind of too tired. But we'll take a 10-minute here before we actually begin. And the next one's, next four, we, 1, 2, 3, 4, we won't take a break. So it's 11. Let's see if we can go till 10. So we got 6 hours. Yeah. Well, yeah. Okay. Move forward. Move forward. We are moving forward, guys. Yes, sir. Yes, sir. You guys even see it? It's kind of blurry for you guys, huh? It's too blurry for you guys. What the heck? Is that better? I think it's better. Yeah, I think it's better now. Maybe you guys can see. Oh, you guys can see, you know? I have to think about you guys too. Yeah. Well, that's a little bit too much. I kind of like this here, showing. Sometimes I do, sometimes I don't. I think I do like it for now. We'll go one more though. Right? Yeah, this is fine. But let me make sure that the chair is angled. Maybe this is better. I kind of like it straight. I think you need to increase the desk by like a little bit. Ooh, that's pretty good. That's perfect. All right, I'm gonna take my 10-minute. Yeah, I'm gonna take my 10-minute break now, and then we'll go and see if we can do the next 4 sections without break. I might read the articles, watch the videos twice for each. First time run through. Second time, we'll do 5x speed, and then we'll see if we have enough time. I need to make sure that I add them to the Anki deck, go over them, and read the high yield as well for it. That way, I can actually catch up. Hey guys, we're back. We're back. Actually, let me finish the other thing real quick before we start. I got some yogurt, by the way. I don't know if you can see. I got some yogurt. I think I'll put the clock back on. That's fine. I'll put the clock. Maybe I'll just take like some breaks. Not too many. Anyways, yeah, let's keep this here and let's put this here. I had it on my phone, but I think it should be fine. Why should I take that long to just kind of quickly do this? Yeah, let's put this over here. You guys don't need to hear my voice so much. Um, Yeah, I think I have it before then. Right? I think so. Okay. Yeah, that's fine. Good. Good. Very good. I think he said what? What did he say? Yeah. Yeah, I'll get it. All right. Let's start. Let's go. We got some food. That's what it's all about. God. Oh my God. Oh my God. Ah, so good. So nice. Stop. 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 An insertion of alanine between us is 543 and 544. That's a gene italicized protein, not italicized. Stop it. Let's go. Rewind the replay.
Mutations. But first, let's go over the central dogma of molecular biology, which is just the idea that genetic information in a
Slow you down,
cell
'cause
is
you're always
stored
too
in the
fast,
form
buddy.
of DNA.
Yeah, slow you down.
And this
Too fast.
DNA is used to generate complementary RNA through a process called transcription. That RNA is then used to synthesize a corresponding protein through the process of translation. So looking at a quick example, our short DNA strand here will be used to generate an RNA strand. Remember that A pairs with
Okay. Continue.
with U or T, and C pairs with G. Next, our RNA will be used to generate protein through translation. And remember that during this process, RNA nucleotides are read in groups of 3, called codons, in order to generate the process of translation. So looking at a quick example, our short DNA strand here will be used to generate an RNA strand. Remember that A pairs with U or T, and C pairs with G. Next, our RNA will be used to, a quick example, our short DNA strand here will be used to generate an RNA strand. Remember that A pairs with U or T, and C pairs with G. Next, our RNA will be used to generate protein through translation. And remember that during this process, RNA nucleotides are read in groups of 3, called codons, in order to generate corresponding amino acids. Now, just very generally, we say that mutations have the effect of making this synthesized protein not turn out quite right. So I'm going to give a quick shout out to sickle cell disease, which is an example of a disease that's caused by a genetic mutation. So you may remember that there is a protein in red blood cells called hemoglobin, which we can also call HB. And hemoglobin is a protein that coordinates iron ions in order to hold on to oxygen molecules and transport them throughout the body. Now, the mutation that causes sickle cell disease results in a mutated form of hemoglobin called HBS being formed, where the S is for the word sickle. And the difference between normal hemoglobin and HBS is that one glutamate amino acid residue is being replaced with a valine amino acid residue. And this small change results in all of these mutated HBS proteins aggregating together in a red blood cell, which makes it very difficult for that red blood cell to transport oxygen effectively. Now, just a side point, remember that red blood cells are initially generated from hematopoietic stem cells through a process called hematopoiesis. So where are mutations found, and how did they come up in the first place? Well, let's look at a couple different possible mistakes that could lead to an incorrectly produced protein. So first, we'll see what happens if a cell makes a mistake during translation, and we'll stick with our example of sickle cell disease from before. So let's say that we have this sample piece of DNA with 3 nucleotides from the gene coding for hemoglobin. This DNA is transcribed to form the complementary RNA sequence GAG. Now that GAG would normally correspond to a glutamate residue during translation, but a mistake during translation might lead to a valine residue being translated instead to produce the mutated hemoglobin associated with sickle cell disease.
Okay.
But notice that if a mutation happens during translation, the cell will only produce 1 mutated hemoglobin, or HbS, for each overall mistake. And since cells are making tons and tons of hemoglobin, just 1 mutated protein might not have that big of an effect on the cell. So we can say that mistakes during translation probably don't cause mutations like the one associated with sickle cell disease. So next, we'll look at mistakes during transcription. Again, we have our CTC piece of DNA, which would normally make GAG on RNA, but maybe a mistake occurs which leads to the transcription of a GUG instead, which would then code for the valine associated with mutated hemoglobin. Now, if this mistake occurred, the cell would only make a few mutated hemoglobins for each mistake, since an individual strand of messenger RNA will only be translated a couple of times before being degraded. So we can say that mistakes during transcription probably don't cause mutations like the one associated with sickle cell disease. Finally, we'll look at mistakes in the DNA strand. If our CTC in DNA is mistakenly turned into a CAC, then our corresponding RNA from transcription will be changed, and ultimately a valine would be produced instead of a glutamic acid. Now, since a cell's DNA stores all of its genetic information, that mistake would lead to all future hemoglobins produced from that gene being mutated. So overall, we can say that mutations will usually result from mistakes in a cell's DNA, and not from the RNA or the protein. So where do these types of mutations come from? Well, there are 2 ways a person can get a genetic mutation. The first is that they inherit it from their parents.
Okay.
Remember that DNA is passed down from parents to offspring. So if we have a mutated father here, then there's a good chance that at least one of his kids will inherit that mutated gene, the same way that the child might inherit any amount of that parent's DNA. The other possibility is that the mutation will come on spontaneously, which is where a person suddenly gets a mutation in their DNA without their parents having had the same mutation. And spontaneous mutations can come from many different sources, with just a few examples being from DNA replication errors, environmental factors like certain poisons, or it's also possible that genetic mutations can come on entirely randomly. So what did we learn? Well, first we learned that mutations originate at the DNA level and not at the RNA or protein level. But the effects of a mutation, like the example we gave of sickle cell disease, are found with problems with the proteins that are ultimately expressed by the mutated DNA. Now, like every rule, there are a couple of exceptions to this one, but we can say that the effects of a mutation are usually found at the protein level. And finally, we learned that mutations are either inherited from a parent or come on entirely spontaneously. Today we're going to talk about the different types of genetic mutations that you can find in a cell. But first, I want to review the central dogma of molecular biology and how the genetic information of a cell is stored in the form of DNA, which is then transcribed to form RNA and then translated to generate protein. Nucleotides from the DNA are transcribed to their complementary forms on RNA, which are then read as codons, or groups of 3, to code for specific amino acids in a larger protein. Now, if you mutate one of the nucleotides on DNA, like let's say turning this thymine base into an adenine base, then that will affect the RNA sequence and ultimately the protein that follows.
Right.
So we say that mutations are mistakes in a cell's DNA that ultimately lead to abnormal protein production. So what are the different types of mutations? Well, the first type of mutations we're going to talk about are called point mutations.
You got it.
Now here I've just written out a random sequence of DNA, which is just a repeating pattern of CTC, which would code for a repeating sequence of GAG in the RNA strand. And finally, a protein sequence of 3 glutamate amino acids.
Okay.
So a point mutation is when one of our DNA bases is replaced with another. So in this example, a thymine base is being replaced with an adenine base, which leads to a change in one RNA nucleotide and ultimately a change in one amino acid. Another type of mutation is called frame shift, which works a little differently. So first, I'll write out the same DNA, RNA, and protein sequences from before, but now instead of changing one base to another, I'm going to add one to the sequence. And here I've thrown in this extra cytosine base that I've written in blue. Now, naturally, this change would lead to an additional guanine base being in the resulting messenger RNA sequence. But what's interesting is that this mutation will change the reading frame of the RNA. Remember that RNA is read in groups of 3, or codons, when being translated to form protein. But now, since we've added an extra G here, all of the codons coming after that extra G will look a little differently. Now, instead of having 3 GAG codons, we've swapped out 2 for GGA codons. This means that 2 of our amino acids in the final protein will be changed.
What?
And in this example,
What are you-
avid or codon.
What are you talking about?
Remember that RNA is read in groups of 3, or codons, when being translated to form protein.
No, let's go back to the
But
beginning.
and frame shift, which works a little differently. So first, I'll write out the same DNA, RNA, and protein sequences from before, but now instead of changing one base to another, I'm going to add one to the sequence. And here I've thrown in this extra cytosine base that I've written in blue. Now, naturally, this change would lead to an additional guanine base being...
Hold on, buddy.
in the resulting messenger RNA sequence. But what's interesting is that this mutation will change the reading frame of the RNA. Remember that RNA is read in groups of 3, or codons, when being translated to form protein. But now, since we've added an extra G here, all of the codons coming after that extra G will look a little differently.
Right.
Now, instead of having 3 GAG codons, we've swapped out 2 for GGA codons. This means that 2 of our amino acids in the final protein will be changed.
Oh, okay.
And in this example, they'll be changed from glutamate to glycine. So you can see that frame shift mutations usually have more significant effects on the final protein than point mutations
Right,
do.
makes sense.
Now, it's important to recognize that both of these mutations are classified and named for how they affect the cell's DNA structure,
Right.
and aren't really named for how they affect the resulting protein.
Okay.
Now, our next type of mutations are nonsense mutations and missense mutations.
That changes the
Let's say we have a
actual-
DNA sequence that normally generates RNA and codes for a cysteine amino acid. A nonsense mutation is any genetic mutation that leads to the RNA sequence becoming a stop
Can you slow
codon
down?
instead.
Slow
Now,
down, buddy.
missense mutations are a little different, and they're any genetic mutation that changes an amino acid from one to another. So in this example, our mutation is changing the resulting amino acid from a cysteine to a tryptophan. Now you can see that nonsense mutations probably affect the resulting protein a lot more than missense mutations do, since that new stop codon that we're creating could chop off a huge section of the protein instead of just changing one amino acid to another.
I guess it depends, though, right? Doesn't it depend on where the stop where it stops at?
So now we can divide the missense mutations even further into a bunch of smaller categories. Silent mutations are when the mutation doesn't actually affect the protein at all.
What? Hold on. Oh.
Since many different RNA codons can code for the same amino acid, it's possible that the mutation might not affect the protein at all.
That's a silent.
So in this example, CCA, CCG, CCT, and CCC in a section of DNA will all end up coding for glycine. So if you change the third base, it wouldn't affect the final protein. Conservative mutations are where the new amino acid is of the same type as the original. So here I have a glutamate and an aspartate, which are both acidic amino acids. So a mutation that swapped out an aspartate for a glutamate would be a conservative mutation.
Gotcha.
Finally, a non-conservative mutation is one where the new amino acid is of a different type from the original. So here we have a serine amino acid, which is a small polar amino acid, being replaced with phenylalanine, which is a large non-polar aromatic amino acid. And this would be an example of a non-conservative mutation, since serine and phenylalanine are different types of amino acids. Now, I'll point out again that all of these mutations are classified and named for how they affect the resulting proteins, and aren't really named for how they affect the cell's DNA. So let's look at a quick example. Sickle cell disease is a disorder.
Slow down now, buddy. Now you're going too fast.
Where hemoglobin, or HB, which is a protein found in human blood, is mutated into a less active form, which we're going to call HBS.
Okay.
And it results from a single glutamate residue being converted into a valine residue. Now we can classify this mutation as a point mutation, since
point mutation, missense mutation, glutamate. Oh, glutamate. That is that's E, negatively charged. Negatively charged, longer chain than aspartate, and that has two double bonded O's, negatively charged, acidic, hydrophilic to valine. Oh yeah, valine.
Only one DNA base is affected, but we can also say that it's a non-conservative missense mutation, since glutamate is being swapped out for valine, and the two are different types of amino acids.
Yes, sir.
Since glutamate is an acidic amino acid and valine is a non-polar one.
Non-charged.
So what did we learn? Well, first we learned that mutations originate at the DNA level, but show their effects on the protein level. And second, we learned that we can classify different types of mutations by either their effects on DNA or their effects on protein.
Okay.
In reference to DNA, we have point and frame shift mutations. And in reference to protein, we have missense
and nonsense.
And nonsense mutations.
Yes, sir. I like this guy. His videos are pretty short all the time. Like always 5 minutes at least, you know? Last one's 4 minutes, so wow.
So today we're going to talk about the causes of genetic mutations.
75.
But first, let's just do a quick review of the idea that mutations are mistakes in a cell's DNA. And there are two main types of mutations that we see when we look at a cell's DNA. And the first is called point mutations. And that's when one DNA base is switched out for another, which usually results in a change to one codon in the RNA sequence.
Right.
Frame shift mutations are when the reading frame of the RNA is altered, and while the actual nucleotides in the RNA sequence haven't changed.
Inserted or deleted frame shift, and then in frame shift. That was called? I'm sorry, in frame mutation. Multiples of 3. Yeah, in frame multiples of 3 amino acids are added or deleted.
Changed that much, the reading frame of the RNA strand has shifted,
Okay.
meaning that many different RNA codons will change as a result.
Right.
And we're going to take a look into what causes these point and frame shift mutations. So point mutations are caused by base substitution, which is when one DNA base is substituted for another.
Right.
And there are a couple of different types of base substitution. A transition is when you have a substitution of adenine for guanine or vice versa, which is a swap between two purines, or a substitution of cytosine for thymine, or also vice versa, which is a swap between two pyrimidines. A transversion is when either adenine or guanine is swapped for either cytosine or thymine. And in this type of base substitution, you have either a purine being replaced with a pyrimidine or a pyrimidine being replaced with a purine. Now, the last kind of mutation that can lead to a point mutation is a mispairing, which some people call mismatching. And that's when a DNA strand has a non-Watson-Crick base pairing.
Transition, transversion.
Of adenine for guanine or vice versa, which is a swap between two purines, or a substitution of cytosine for thymine, or also vice versa, which is a swap between two pyrimidines. A transversion is when either adenine or guanine is swapped for either cytosine or thymine. And in this type of base substitution, you have either a purine being replaced with a pyrimidine or a pyrimidine being replaced with a purine. Now, the last kind of mutation that can lead to a point mutation is a mispairing, which some people call mismatching. And that's when a DNA strand has a non-Watson-Crick base pairing.
So
Normally,
A doesn't go to T and C doesn't go to G.
A pairs with T and G pairs with C. But when you have a mispairing, that's when A and C pair up, or when G and T pair up. And it's much more common for mispairings to occur between a purine and a pyrimidine, as opposed to between two purines like A and G pairing up, or two pyrimidines like C and T pairing up.
Okay.
Next, we're going to talk about frame shift mutations. So let's say that we have this DNA strand here with 3 repeating CTC units and an extra C on the end.
Okay.
This would then be transcribed into an RNA strand with repeating GAG units and an extra G on the end. And our 3 codons would be the 3 GAG units, which would then each translate to a glutamate amino acid. Now, one way you can cause a frame shift mutation is through an insertion. And that's when an extra DNA base finds its way into our sequence. So here we have this extra cytosine base that I've underlined falling into our sequence. And this additional C base would lead to an extra G being thrown into our RNA sequence, which would then shift the codon reading frame of our RNA strands during translation. So now, instead of 3 GAG codons, we have just 1 GAG codon and 2 GGA codons with 2 extra bases on the end.
Right.
This would then code for 1 glutamate residue and 2 glycine residues instead of 3 glutamates. The other way that you can cause a frame shift mutation is through a base deletion. So in a deletion, we drop off one of our bases from our original sequence. So here I've dropped that first thymine base. And this would also result in a shift of the RNA reading frame. Now, instead of having 3 GAG codons, we have a GGG codon and 2 AGG codons, which would lead to a protein with a glycine and 2 arginine amino acids. So overall, insertions and deletions can both lead to frame shift
What did you
mutation.
do? What did you do? 3, 6, 9, 10. Oh, I see. So he deleted this T here. T, TCC, TCC. Okay.
Now, we can also talk about large
Go backwards,
overall also result in a shift of the RNA reading frame. Now, instead of having 3 GHG codons, we have a GGG codon and 2 AGG codons, which would lead to a protein with a glycine and 2 arginine amino acids. So overall, insertions and deletions can both lead to frame shift mutation.
Yes, sir.
Now, we can also talk about large-scale mutations, which instead of being at the level of individual nucleotides, are usually seen at the chromosomal level and can affect many genes instead of just a few base pairs. So first, we'll talk about translocation, which is when a gene from one chromosome is swapped for another gene on a different chromosome. Now, it's important to see that translocation refers to gene swapping between non-homologous chromosomes, which means that if this blue chromosome were chromosome 10, then the green one could be any chromosome aside from chromosome 10. And this is what sets translocation apart from the process of crossing over that occurs during meiosis between homologous chromosomes. The next large-scale mutation we'll talk about is
Go back.
nucleotides are usually seen at the chromosomal level and can affect many genes instead of just a few base pairs. So first, we'll talk about translocation, which is when a gene from one chromosome is swapped for another gene on a different chromosome. Now, it's important to see that translocation refers to gene swapping between non-homologous chromosomes, which means that if this blue chromosome were chromosome 10, then the green one could be any chromosome aside from chromosome 10. And this is what sets translocation apart from the process of crossing over that occurs during meiosis between homologous chromosomes. The next large-scale mutation we'll talk about is chromosomal inversion, and that's when 2 genes on the same chromosome switch places. So here, our green and blue genes are being swapped and end up on different parts of the chromosome after the mutation. Now, since both of these mutations don't always affect the individual nucleotides coding for a gene, it's important to see that many of these types of mutations affect how a gene's expression is regulated, in addition to changing what the genes actually code for. Remember that the position of a gene on a chromosome partly determines how it's regulated, and this could be due to histone configuration, promoter regions, or any other regulatory process. So what did we learn? Well, first we learned that small-scale mutations affect DNA at the nucleotide level. And of these small-scale mutations, we have point mutations, which can be caused by transitions, transversions, and mispairings. And we also have frame shift mutations, which can be caused by insertions or deletions. Next, we talked about large-scale mutations, which affect DNA at the chromosomal level. And the 2 large-scale mutations we talked about were translocation and inversion.
All right, buddy.
So today we're going to talk about mutagens and carcinogens and how they can cause DNA damage. But first, I want to review the central dogma of molecular biology and how the genetic information of a cell is stored in the form of DNA, which is then transcribed to form RNA and then translated to generate protein. Now, nucleotides from DNA are transcribed to their complementary forms on RNA, which are then read as codons, or groups of 3 to code for specific amino acids in a larger protein. Now, if you mutate one of the nucleotides on DNA, like turn this thymine base into an adenine base, then that will affect the RNA sequence and ultimately the protein that follows. So we say that mutations are mistakes in a cell's DNA that ultimately lead to abnormal protein production. So what is a mutagen?
Right.
Well, a mutagen is any chemical substance or physical event that can cause genetic mutations. Chemical substances like certain poisons could be mutagens, or physical events like UV light will event that can cause genetic mutations. Chemical substances like certain poisons could be mutagens, or physical events like UV light or different kinds of radiation could also be mutagenic. And we classify mutagens into 2 different categories. So let's say we have a person over here. A mutagen could be classified as endogenous, and it comes from inside this person's body. and it's some mutagen that's already found in the organism. But an exogenous mutation is one that comes from outside the affected organism, something that's from the external environment. So what are some examples of some endogenous mutagens? Well, the most significant endogenous mutagens are what we call reactive oxygen. Well, the most examples of most significant endogenous mutagens are what we call reactive oxygen species, or ROS.
Yeah, talking slow now.
And ROS are naturally occurring metabolites in the human body that are produced by mitochondria during oxidative phosphorylation. So if we have this guy here who's about to chow down on a big meal, you can expect that during the metabolism of the meal, his mitochondria will produce a bunch of ROS, like O2-, which we call superoxide, which is an oxygen molecule with one extra electron, as well as some hydrogen peroxide, which is another ROS that your body can produce. Now, reactive oxygen species, as you may be able to tell by their name, contain oxygen, like both of these examples do, but they're also highly reactive with different cell components, including DNA. And by reacting with DNA, they can actually cause significant damage to a cell's genetic code. One example of this type of damage is the double-strand break. And ROS can actually break a DNA's double helix into two smaller pieces.
Oof.
And you can see why this type of a reaction could cause a mutation, since it quite significantly changes the structure of the cell's DNA.
Yep.
The next type of DNA damage that ROS can cause is base modification. And that's when the nucleic acid bases are changed or swapped around, and that can pretty readily cause point mutations, or maybe even other kinds.
Okay.
Now, you may be wondering, why would a cell ever make something that could damage itself?
So I just
What?
ate a meal. I'm causing endogenous mutations within my body.
Turns out that ROS actually have a couple of beneficial effects on a cell. And cells actually have a couple of ways to make sure that they don't cause damage. But sometimes ROS levels get really high and cells can't deal with them anymore. And we call this oxidative stress.
Okay.
And antioxidants are something that your doctor might have told you they're good for you. And it turns out that part of what antioxidants do is help make sure that ROS don't damage your DNA.
I think I have enough antioxidants.
Now let's look at a couple examples of exogenous mutagens. And there are many different types of
Really?
exogenous mutagens, but we're
X-ray?
really only going to talk about two. Now, intercalators are one example, and
Okay.
one of them is called ethidium bromide, which you may be familiar with if you've ever done a PCR experiment before. And what ethidium bromide will do is it'll jump into a DNA double helix and stick itself between the two strands. And when these intercalators intercalate into DNA, they can deform the structure of the DNA and cause some serious problems. Base analogs like 5-bromouracil, which we also call 5-BU, pretend to be a certain base, but then act differently than that base normally would. So in the case of 5-BU, it's an analog of uracil and looks a lot like it, but once it's incorporated into DNA, it can shift between two different forms.
Whoa.
In its keto form, it will pair best with adenine, while it's in enol form, it will pair best with guanine.
Oh.
Now, if you're familiar with organic chemistry, you might know that 5-BU can convert between its keto and enol form through something called a tautomerization reaction. And overall, you can see how this base analog might be able to induce mutations in a DNA strand. Now, the last thing we're going to talk about is what a carcinogen is. Now, carcinogens can be mutagens, but not all of them are. But in general, you can say that a carcinogen is something that can lead to cancer, which, if you remember, is when cells in an organism divide uncontrollably and can form big masses of cells called tumors.
Yep.
Now, some carcinogens will work by making mutations in DNA that lead to cancer, but sometimes they might carry out their effect simply by increasing the rate at which a bunch of cells divide without actually affecting their DNA. And some examples of carcinogens are tobacco, which come from cigarettes, asbestos, which used to be used as home insulation, and even UV radiation. So what did we learn? Well, first we learned that mutagens are chemical or physical substances or events that can increase the probability of genetic mutations occurring. And next, we learned that carcinogens are things that lead to cancer. And while they can be mutagenic as well, they aren't necessarily mutagenic.
All right.
So today we're going to talk about the overall effects of a genetic mutation and how mutations impact the affected organism as a whole. But first, I want to review the central dogma of molecular biology
Okay.
and how genetic information in a cell is stored in the form of DNA,
Let's do it, buddy.
which is then transcribed to form RNA, which is then translated to form protein.
right?
Now, nucleotides from DNA are transcribed to their complementary forms on RNA, which are then read as codons, or groups of 3, to code for specific amino acids in a larger protein.
Okay.
Now, if you mutate one of the nucleotides on DNA, like turn this thymine base into an adenine base, then that will affect the RNA sequence and ultimately the protein that follows. So we say that mutations are generally mistakes in a cell's DNA that lead to abnormal protein production. So are mutations good or are they bad? And what kind of effect do they have on the affected organism? Well, there isn't really a good answer to this question at all. And there are many, many different types of mutations out there that can result in big structural changes, like the little pictures I've drawn out here, or may result in little subtle changes that might go completely unnoticed. It's very difficult to call a mutation good or bad, though, since it really depends on a huge number of things, including the environment that the organism lives in.
Okay.
So let's look at an example of a good mutation. So the bacteria Streptococcus pneumoniae is the bacteria that you typically see associated with pneumonia. And one of the more popular treatments for pneumonia is giving the infected person an antibiotic like penicillin, which would help kill all of the bacteria and get rid of the disease. But sometimes you can find some mutated Streptococcus bacteria that will have a special trait that makes them resistant to penicillin. And now penicillin won't kill them as easily as it'll kill the bacteria without the mutation. Now, we call this a good mutation because the bacteria are living in a human host where they're likely to encounter this deadly penicillin. And being resistant to antibiotics like penicillin would then be beneficial to the bacteria. And just to clarify, I'm calling this a good mutation for the bacteria,
not for us.
Not really for the human infected, since
Yeah.
it'll be harder for them to get rid of the bacteria that are resistant to certain antibiotics.
Right.
So now let's look at an example of a bad mutation. So the disease cystic fibrosis is usually caused by a mutation in the CFTR gene. Now, I'm not really going to go into detail about how this mutation actually hurts you, but I'll leave you with the idea that what it does is it makes the mucus that you'd find in a person's lungs really, really thick,
Gotcha.
which makes it really hard for people affected with the disease to breathe.
Okay.
So in general, we can say that the mutation causing cystic fibrosis would be a quote-unquote bad mutation. But mutations aren't strictly good or strictly bad. In fact, there are some mutations that can cause some favorable and some disadvantageous effects. Sickle cell disease results from a mutation in a protein called hemoglobin that you'd find in red blood cells. And this mutation turns hemoglobin into a much less functional form, which we'll call HbS. And it's much less efficient at moving oxygen around the human body.
Okay.
But another effect of sickle cell disease is that it makes the diseased person less susceptible to malaria. Now, malaria is a parasite that grows and multiplies in red blood cells and can have a lot of nasty effects on the host organism. And the malaria parasite can't really grow as well in red blood cells that are affected with sickle cell disease. So, in this case, the mutation associated with this disease has one bad effect, which is that the HbS isn't as good as carrying oxygen around the body, but also a good effect in that it makes it less likely that the diseased person will be affected by malaria, since they can't grow as well in the human's red blood cells. So what did we learn?
Okay.
Well, first we learned that the effects of the mutation will usually, but not always, appear at the protein level. There are some exceptions to this rule. And second, we learned that genetic mutations can have advantageous, deleterious, or neutral effects, depending on the type of mutation, the environment that the affected organism lives in, as well as a multitude of other factors.
All right, thanks, bud. Let's go ahead and start the questions. Yeah, I'm ready to practice. That's what he said, from what I remember. That'd be a point mutation. Conservative? Is it conservative? Dude, I don't know the codon for glutamine and valine. Are you kidding me right now? It wasn't deletion. Well, but it's okay, so it's glutamine. Is it glutamine? Well, no, GLU is glutamate. Oops. GLU is glutamate. Glutamate is acidic. Valine is, yeah. I'm going to say it's no, wait, he said conservative. Conservative was if it's, if they just changed it to a similar AA. This one is a frame shift. Frame shift, in frame, frame shift. No, it's this one then. Yeah, let's go. Stop. Highly unlikely to affect protein because so a single codon codes for one amino acid, but an amino acid can be coded for by multiple codons. Multiple codons can code for the same amino, which is the same thing we're saying, right? In fact, I don't think that's true though. That was it. I'm not sure about that. This one. Not conservative mutation. It's pretty. They're dissimilar. They're not very similar. New amino acid is different. Yeah. Let's go. A transition. Oh man, I forgot. Somebody said transition. I think that one's like if it's different. A goes to T, so that's normal. A goes to T, that's normal. G goes to A. I think this one. Oh, let's go. Purine substitute for another purine, or purine substitute for purine. Oh, okay. Large scale deletion, so in frame mutation. Oh. The nucleotide level mutations. Large scale deletions. I mean, this one makes sense. I mean, that's what I'm thinking about it doing. DNA. At which level? In frame. Let's find out the nucleotide levels. Yeah, maybe. I think it's at the translation. At the point of translation. So I think this one. Then it's D. Oh, okay. I'll check that check. Right. Could reduce methylation. I don't know. Large scale deletions. Well, what are large scale deletions? I guess that makes sense. Frame shift is a large scale deletion. Large scale deletions. Oh, large scale deletions at the chromosomal level. Oh, frame shift mutations. What is this one then? Oh, that's part of the insertion or deletion. Frame shift mutation happens at the chromosomal level. Oh man, I got that mixed up. So frame shift is large scale, happens at large scale. Insertion deletion, smaller scale. I think mRNA and point mutations also. Point mutations and frame shift mutations both happen in mRNA. Frame shift Or I think bullshit. I'm sure happens in there. Y scale deletions. I think this is at the chromosome level. Yeah, okay, I was thinking I need to zoom out on that one. Okay, nitric oxide is part of this cardiovascular anyway. Because in DNA mutations we occur in cells. These mutations are not always associated with cancer formation. Well, it's definitely endogenous. It's not always associated with cancer, so it's mutagen. What is the benefit of antioxidants? So can O2. This one. Intercalation of lithium bromide results in deformation of DNA. Research studying immunity in a gene associated with muscle function. So P glycine at the 12th amino acid position is exchanged for a valine. Glycine is exchanged for a valine. That's I mean, like this one had a hydrogen, this one has a methyl group. The defective protein loses the ability to bind calcium ions. Well, that makes sense. I mean, it doesn't have a really readily available hydrogen group to donate. Loss of hydrogen bonding, which is critical for protein bonding. Is it critical for protein bonding? That's not true for protein. Oh, for protein folding. Protein folding? That's not true. That's not true at all either. So we have B or A. They make sense. Binding pocket for calcium. Bonding capacity at the N terminus. At the N terminus. Does the N terminus still have? The N terminus still has this hydrogen. Yeah, I'm gonna say A. He still has it. The next screen identifies C. C. So the coding strand. The coding strand. Deletion between codon. Wait, TTC. Maybe 3, 4, 5. 3, 4, 5. Okay, a deletion of a TTC. Okay. It's going to be what it is, right? Loss of integrity of the cell membrane. TCC at the coding strand. So that's going to be G AG, which is wait. No, I think it's just that. This one? Okay. Gene has the following original DNA sequence, permutation sequence of 2. Okay, what was that? Post mutation, ATG, AAG, GTTGA. So it's an insertion, which missense, which would more likely cause a missense. Frame shift. I have to actually try and do it, I guess. ATG, AAG, GTT, GTT, G. Oh, wait. ATG, AAG, AAG, GTT, TGA. UAC. CAA. ACU. Now with the other one, what do we got? We got ATG, ATG, AAG, GTT, and GA. And so we lose an amino acid here. I think it's going to be it's not a point, so that's a frame shift. And that's just an altered protein sequence because it doesn't have the other amino acid that it was going for originally. Okay. Oh, okay. In frame. It's also in frame mutation, not just a frame shift. Two mRNA sequences from the same gene shown from below. So this contains stop codon. Okay. mRNA1. Well, I mean, I'm just looking for. I mean, this. I'm just looking for Uga, right? Okay, Uga's here, and then Uga's here. So sequence one has 1, 2, 3. It's 3, and then sequence 2 has 1, 2, 3, 4, 5, 6. So 3 and 6, I'd say. Let's go. Well done. 6 questions. Ugh. Okay. God, there's most abundant protein in mammals. With type 1 collagen being the most prevalent. Type 1 is found in bones and ligaments, relatively 3 plant chain, which wind around one another in a triple helix, stabilized by covalent cross linkages. Others is limit are composed of regular type or X, but this can be any number. Okay. Osteogenesis affect typically is also a genetic mutation in the collagen genes. Type 1 in the COL1A1 gene production. The types of osteogenesis perfect includes 2, 3, 4, 2, 3, 5. I think it's 2, 3, no, 2, 3, 4. From bones that fracture easily. What is the mutation that causes an effect to first occur? Genetic mutation, it just said. Right? Am I messing around? Connective tissue. Collagen gene. What the heck? What the heck? With collagen retreated at the protease X, with protease X, which cleaves the carboxyl glycine, which cleaves the carboxyl terminal. Just one. We just have one left. Right? So we got rid of the glycine and we got rid of the proline. We just have that X. Oh no. Oh, that's not what it's saying. Cleaves at the carboxyl terminal of glycine. The carboxyl. Okay, so you got rid of the glycine. Amino of proline. The amino of proline. Oh no, you have two. You still have proline and you still have the X. Oh, what the heck? What are we talking about here? Okay. Amino terminal. Proline. You have two residues left, right? What? How's it three? Oh, what? This leaves a three long residues long for him. I guess that's a good question for me to look into. Huh? Oh, I didn't know that. Oh, that was quite critical information right there, huh? Where'd it go? Three polypeptide chains. Gotcha. Okay, GlyPro X will cleave at the C terminal of Gly. So we got Pro X Gly Pro. Why do we have X Gly Pro? It cleaves at the terminal. It cleaves at the terminal. Which cleaves at the carboxyl terminal, amino terminal. Protease X cleaves at the carboxyl terminal glycine. Carboxyl terminal glycine is here. Yeah, GlyPro. It cuts after glycine. What? if we cleave after glycine, let me see if I can figure this out. Cleave after glycine. Okay, and then we also do what? At the amino and proline. Yeah, the first one is taken out. That's what I said, dude. That's literally what I said. Oh. Oh, actually, so Pro X is not left out. Gotcha. Oh, wow, that's pretty cool. Yeah, okay, so Gly here is gone. We have Pro X Lite, and Pro X. Okay. Oh, dude. I need to move on to the other ones, but I do want to do it. Kind of in a tough position. The most common form of influenza type 1 arises from dominant mutation in the CO1 gene. Deficiency. So patient B has the CO1 gene, and the rest. I think patient B, patient 1 and 2. alter the structure of type 1. Patient B and D, right? I don't know. Well, oh, C is also lower than the control. Patient B and C. Okay. Oops, I didn't see that. How many more questions? Oh my god. Which of the following symptoms go through? It's inherited, right? Both parents are recessive. It's from genetic mutation. Dominant. Dominant, so it's only coming from one parent. One allele was mutated so that the one motif was now TRP Pro X or XMU. What disease type? So it was what before? Glycine, now it's tryptophan? That's a pretty huge difference. I think it's a pretty huge difference. Would it be lethal? Well, X can be any time, but it has to be gly. Type 4? Well, we don't know. Dominant mutations. Mutations that alter the gene structure. Maybe they could have a different one, right? Not gonna have type 1. Not gonna have type 1. I don't know if it's gonna be lethal or not. Just have type 4. This is well. Yeah. Oh, okay, yeah. Resulting in only nonsense. Frame shift. What are you missing? Okay. Oh my goodness, dude. Is this the same for the other rest? What happened? Oh man. It's an article? A question. There's more questions. 2 questions. One question, that's not too bad. 3 questions. Jesus. I'm not doing it. DNA, meiosis. 2 questions. 2 questions. 1 question. Alright, whatever. Let's continue. We might as well try. Related content? All the recessive. Maybe multiple allele inheritance. Okay. Complete dog, incomplete. In the species of blue feathers, are completely dominant to the gene for Y fillers. Blue fillers. White bird. 50%. 25%. I don't know. 20%. 0%. I'm just gonna skip this one, dude. Yeah. We can do this later. We'll come back to this. Like I have to relearn all this stuff. This is for the next section, so I don't know why it's giving it to me here. But whatever. I'm wondering if I should add it now. If I should add it now. I think I should. Yeah, let's try to add it now. Sure. Okay, so 8 was done. Pretty nice. It was pretty nice. Now we're on here. Oh, it's the same dude. Let's go. Ooh, a lot of reading now. Is this the one that's next? Is this the one that's next? Or... Let's check, because I'm not sure. Where's my phone? I think. That was one of some related concepts. Let me send this to something. Oh, what the heck? How do I send this to myself? I think it's these two. Meiosis and other meiosis and other spiders. Let me see. Yeah, mentality limits, chromosomal inheritance. Mentality limits. I guess the question is bioenergetics. Oh, okay. Yeah. So it is on there. What is this then? Ah, okay. This is just kind of like a introduction to it, somewhat. Oh, sure. I guess we can add this one here then, right? Okay. Anyways, what's next? Mendelian genetics. So, Mendelian genetics, which you're here. This is your Mendelian genetics. Okay. That. But now let's add to the Yankee cards. Real quick, the mutation section. That looks good. Not here yet though. Oxidation reactions. What do you say? Yeah, I think that's fine. That makes sense to add. I'll add you up to here. Okay. If I type in mutation annotations. Mutation annotation? Nope. Okay. Genetic mutation. That's it. That's really it. Well, maybe add this one. But types of mutation. Hypermutation? No, I'm not there yet. What? Why did I move that down? Oh, the causes. That's already there. Mutagens. Mutagen. Okay. Here we go. There's a little bit more here. Oh my goodness, what the heck? That's a lot. Ha 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 ha. I think I got it. Yeah. Yes. 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 Yep. 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 Biology. Yeah. I think this would be part of it as well. This one, yep, also part of it, but not this one. I don't know in there. Maybe just this one. Or that one. Yep, these ones for sure. Bacterial Oh, not this one. Transposing. No, not you. Get out of here. Get out of here. Maybe that's probably what the next one that Italian. Oh, actually, what's a? Oh, that was it. Just the mutations, huh? Yeah. Okay. Next one. Box you up. And then that's added. So exit. Sync. Oh, I think it doesn't matter if I add them today. I already did the ones for today. It's not gonna add any more, huh? Oh, that's so nice. Oh, that's great. Well, let's put you down. We will do this one here today. That one will be for lesson 10. But it wants me to hit DNA technology after, which doesn't really make any sense. Really, I shouldn't need that. Whatever. I'm going to do somebody else's DNA. That's already it. Evolution. Anyways. We might make it. I don't know. Maybe we'll do 3. I mean, yeah, let's see how much we can do. But we also have to read the IFDU high yield. Data genetics and related concepts has a little bit. Can I? I can't. Okay, the law of segregation. Law of independent assortment. Codominance. Codominance over predominance. Punnett squares. Probabilities. In genetics, pleiotropy and lethal alleles. Pleiotropy. Alleles, variations on Menger's law. All right. Well, yeah, that's good. Let's refresh this. Let's put in lesson 10. Which over there. That's lesson 10. Yeah, headland. 8 and 9. 10, because that's the way it is here. DNA technology. Oh, DNA technology and then chromosome. I feel like doing them down genetics and then chromosomal would make sense. So we're actually getting this. I'm gonna do it that way. DNA technology. Clone genes. Expression function. I think that makes sense to me. It makes sense to me. Makes sense to me. Huh? This makes sense. Doing this way. 9, 10, and then DNA. Wait, is that what you do? Oh, that's the next one. That's way after that. Yeah, this is fine. I'm gonna do this one. Alright, but for now, let's take a quick 10-minute before we actually hit the next section. We might make it today. I don't know. We'll see. Depends if I have any hiccups. Now it's been okay. 2 out of 4. Yeah, that's fine. Excuse me. Excuse you. You need to leave. You need to leave. Excuse me. No. You need to leave. What did you say? I can't believe you've done this. Oh, actually, no, I like the blue pens better. You need to leave. Let's go, Mendel. Let's go, Mendel. I say you're walking through, and then we do 2x speed. What are you doing? What are you doing? Get out of here. Go on, get. And let's see. Right? Well, I mean, it looks like it's only a little bit, but I gotta read it through. How long are these? Oof. Oof.
I just-
Oh man, no, these are too long. Might not make it. But we're gonna try. Why did it start me there when I have to start here? Okay. Go here.
An introduction to Mendelian genetics.
Oh, this guy's quick. Oh, and then just a bunch of. Okay, reading. Let's go. Start the clock. Mendelian genetics. Mendelian genetics and related concepts.
Now, before we start, let's review the idea that human cells contain 46 chromosomes, which contain the DNA that makes each cell unique. And 23 of these chromosomes were inherited from a person's father, and 23 were inherited from the mother.
Okay.
So we can say that each person is made up of a combination of genetic code from both of their parents. Now, sometimes we like to say that we have 23 pairs of chromosomes instead of saying that we have 46 total, because that way we remind ourselves that for each chromosome we have a maternal and paternal copy. Now, the first thing I want to is we have 23 pairs of chromosomes instead of saying that we have 46.
Okay.
6 total, because that way we remind ourselves that for each parent's week, and 23 of these chromosomes were inherited from a person's father, and 23 were inherited from the mother. So we can say that each person is made up of a combination of genetic code from both of their parents. Now, sometimes we like to say that we have 23 pairs of chromosomes instead of saying that we have 46 total, because that way we remind ourselves that for each chromosome we have a maternal and paternal copy. Now, the first thing I want to introduce is the term allele. Now, if we have a chromosome here, then an allele is one small section on that chromosome that codes for a specific gene that makes you, you. Now, since humans introduce is the term allele. Now, if we have a chromosome here, then an allele is one small section on that chromosome that codes for a specific gene that makes you, you. Now, since humans have at least 2 copies of each chromosome, we can say that humans usually have at least 2 alleles for every specific gene, 1 allele from their mother and 1 from their father. So let's look at an example, and we'll start by talking about blood type. Now, I'm sure that you've heard that blood types are usually named with letters like A, B, and O. But what does that actually mean? Well, there's a specific allele that codes for blood type. And let's say that we have this guy here, and his alleles both code for blood type A. And I'll use the letter A for that. And let's say we have this girl here who has one allele coding for A and another allele coding for blood type O. Now for the guy, if he has both alleles coding for blood type A, then it's pretty clear that when we check his actual blood type, it will be A. But for the girl, we're not so sure since she has one of each. So now I'm going to introduce a couple new terms to you. And the first is that since the guy has 2 alleles that code for the same thing, both code for blood type A, then we say that this guy is homozygous. And homo means the 2 alleles are the same, homo the same. And zygous refers to the mixture of DNA that he got from his parents. So someone who is homozygous got the same allele from both parents. Now, in the case of the DNA that he got from his parents, So someone who is the two alleles are the same, homo the same. And zygous refers to the mixture of DNA that he got from his parents. So someone who is homozygous got the same allele from both parents. Now, in the case of the girl, is she going to have type A? And I'll be clear that when we check his actual blood type, it will be A. But for the girl, we're not so sure since she has one of each. So now I'm going to introduce a couple new terms to you. And the first is that since the guy has two alleles that code for the same thing, both code for blood type A, then we say that this guy is homozygous. And homo means the two alleles are the same, homo the same. And zygous refers to the mixture of DNA that he got from his parents. So someone who is homozygous got the same allele from both parents.
Gotcha.
Now, in the case of the girl, is she going to have blood type A or blood type O? Well, it turns out that she's going to have blood type A, and that's because the A allele is the dominant allele, while the O allele is the recessive allele. When an allele is dominant, that means that if someone has two different alleles, it'll be the dominant one that wins. So in this case, since A is dominant over O, which is recessive, A will win, and she'll have blood type A. Now, since this girl has two different alleles, we call her heterozygous, since hetero means different, and zygous refers to the same thing, the mixture of DNA that she got from her parents. Now, I want to introduce two more terms. We can describe a person's genes in two different ways. We can look at the person's individual alleles, and we call this the genotype. So for this guy, his genotype is AA, referring to his two alleles, which both code for blood type A. But we can also look at a person's physical traits, which we call the phenotype. So for this guy and girl, the phenotype would be blood type A. So you can see that genotype and phenotype are different, but it is possible for two different genotypes to make the same phenotype, since some alleles are dominant over others. Now let's talk about gene inheritance for a bit. So let's say that our guy and girl from before have offspring together. We can use something called a Punnett square to determine what different genotypes their kids could have. Now, each of the parents' two alleles are on separate chromosomes, so each parent will contribute one of their two alleles to the child, and the Punnett square allows you to determine all possible combinations. So if we take the father's alleles and line them up vertically, and then take the mother's alleles and line them up horizontally, we can fill in the chart to find the possible genotypes for our offspring. In this case, two of our boxes will have the AA in them, and two will have AO in them. So that means that half of the children will have the genotype AA, and half of the children will have genotype AO. But since both of these genotypes code for the same phenotype, all of the children will have the blood type A phenotype. Now let's see what happens if we change our father's genotype to match our mother's genotype. Now, only 1/4 of the children will have the AA genotype. Half will have the AO genotype, since the order
of 25% right now. Oh yeah.
of the two alleles doesn't matter. OA and AO are the same. And 1/4 AA, and line them up. Now, each of the parents' two alleles are on separate chromosomes. So each parent will contribute one of their two alleles to the child. And the Punnett square allows you to determine all possible combinations. So if we take the father's alleles and line them up vertically, and then take the mother's alleles and line them up horizontally, we can fill in the chart to find the possible genotypes for our offspring. In this case, 2 of our boxes will have the AA in them, and 2 will have AO in them. So that means that half of the children will have the genotype AA, and half of the children will have genotype AO. But since both of these genotypes code for the same phenotype, all of the children will have the blood type A phenotype. Now let's see what happens if we change our father's genotype to match our mother's genotype. Now, only 1/4 of the children will have the AA genotype. Half will have the AO genotype, since the order of the two alleles doesn't matter. OA and AO are the same. And 1/4 will have the OO genotype. So this means that 75% of the children will have blood type A in their phenotype. phenotype since AA and AO make blood type A, but 25% of the children will have the blood type O phenotype.
There's a 25% chance a child of this cross will have the phenotype O.
Since OO makes blood type O. So what did we learn? Well, first we learned what an allele is, and the difference between homozygous and heterozygous, as well as the difference between dominant and recessive traits in relation to alleles. Second, we learned about the difference between genotype and phenotype, and how the genotype refers to a person's DNA, while a phenotype refers to the physical traits that the DNA codes for. And finally, we learned about how we can use a Punnett square to determine how different alleles will be inherited from two parents.
Okay, let's get to it. Let's get to learning about some peas. Good writing.
2 copies of each. Oh, at least is the we like to believe that. A couple who's someone who is homozygous got dominant. That means that if someone has 2 different alleles, it'll be the dominant one that wins.
Oh, gotcha. Only one of the two copies of genes is distributed to each gamete, and the allocation of the copy is random. It's random, but it can be random if the parent is dominant, right? Like if that same allele on that gene two gene copies. Two pure breeds. When you cross all of the F1, all of the F1, all of the F1 are going to be Y. I mean, I guess they're going to be BT, right? I guess that means that the tan is going to be dominant while black is recessive. What makes the other one recessive? function. Ah, gotcha. It's because it only needs one copy to actually show its phenotype. Well, maybe for the recessive it needs both, and that's why it's dominant. Right? Because this is y, but this is y, lowercase. Okay. So that means that someone must have had blue eyes in the past, right? Sure, anyways, let's keep on. You're inherited independently or not. mean, this one we just did. But maybe this one.
We even knew what DNA was, much less how it was structured or was replicated, or even before we could look in and see meiosis happening in cells, we had the general sense that offspring were the products of some traits that their parents had. You know, if I had a guy with blue eyes, say this is the blue-eyed guy right here, and then if he were to marry a brown-eyed girl, say this is the brown-eyed girl, maybe make it more like a girl. If he were to marry the brown-eyed girl there, that most of the time, or maybe in all cases where we're dealing with a brown-eyed girl, maybe their kids are brown-eyed. So they have a little brown-eyed baby here. Right? And this is just something, I mean, there's obviously thousands of generations of human beings, and we've observed this. We've observed that kids look like their parents, that they inherit some traits, and that some traits seem to dominate other traits. And one example, that tends to be a darker pigmentation in maybe the hair or the eyes. Even if the other parent has light pigmentation, the darker one seems to dominate, or sometimes it actually ends up being a mix, and we've seen that all around us. Now, this study of what gets passed on and how it gets passed on, it's much older than the study of DNA, which was really kind of discovered in the, or it became a big deal in the middle of the 20th century. This was studied a long time, and kind of the father of classical genetics and heredity is Gregor Mendel. Gregor Mendel was actually a monk, and he would mess around with plants and across them and see which traits got passed and which traits didn't get passed, to try to get an understanding of how traits are passed from one generation to another. So when we do this, when we study this, classical genetics, I'm going to make a bunch of simplifying assumptions, and because we know that most of these don't hold for most of our genes, but it'll give us a little bit of sense of how to predict what might happen in future generations. So the first simplifying assumption I'll make is that some traits have kind of this all or nothing property, and we know that a lot of traits don't. Let's say that they're in the world, and this is a gross simplification. Let's say for eye color. Let's say that there are two alleles. Now remember what an allele was. An allele is a specific version of a gene. So let's say that you can have blue eye color, or you could have brown eye color, or you could have brown eye color. And we live in a universe where someone could only have one of these two versions of the eye color gene. We know that eye color is far more complex than that. So this is just a simplification. And let me just make up another one. Let me say that, I don't know, maybe for tooth size. That's a trait you won't see in any traditional biology textbook. And let's say that there's one trait for big teeth, and that there's another allele for small teeth. And I want to make very clear this distinction between a gene and an allele. So let's I talked about Gregor Mendel, and he was doing this in the 1850s, well before we knew what DNA was, or what even chromosomes were, and how DNA was passed on, etc. But let's think a little bit about, let's go into the microbiology of it to understand the difference. So I have a chromosome. Let's say on some chromosome, let me pick some chromosome here. Let's say this is some chromosome. Let's say I got that from my dad, and on this chromosome, there's some location here. We call that the locus on this chromosome, that where the eye color gene is. That's the location of the eye color gene. Now I have two chromosomes, one from my father and one from my mother. So let's say this is the chromosome from my mother. And we know that when they're normally in the cell, they aren't nice and neatly organized like this in a chromosome, but this is just going to show you the idea. Let's say these are homologous chromosomes, so they code for the same gene. Now, there's some location here, we call that the locus on this chromosome, that where the eye color gene is. That's the location of the eye color gene. Now, I have two chromosomes, one from my father and one from my mother. So let's say this is the chromosome from my mother. And we know that when they're normally in the cell, they aren't nice and neatly organized like this in a chromosome, but this is just going to show you the idea. And let's say these are homologous chromosomes, so they code for the same genes. So on this gene from my mother, on that same location, or locus, there's also the eye color gene. Now, I might have the same version of the gene, and I'm saying that there's only two versions of this gene in the world. Now, if I have the same version of the gene, I'm going to make a little shorthand notation. I'm going to write big B, actually, let me do it the other way. I'm going to write little b for blue, and I'm going to write big B for brown. This, there's a situation where this could be a little b, this could be a big B. And then I could write my genotype. My genotype, I have the allele, I have one big B from my mom, and I have one small b from my dad. Each of these instances or ways that this gene is expressed is an allele. So these are two different alleles. Write that. Two different alleles or versions of the same gene. And when I have two different versions like this, one version from my mom, one version from my dad, I'm called a heterozygote, or sometimes it's called a heterozygous genotype. Zygous genotype. And the genotype is the exact versions of the alleles I have. If I had, let's say I had the lowercase b, I had the blue-eyed gene from both parents. So let's say that I was lowercase b. Then I would have two identical alleles. Both of my parents gave me the same version of the gene. And in this case, I'm called, this genotype is homozygous. Or this is a homozygous genotype, or I'm a homozygote for this trait. Homozygotes. Now you might say, Sal, this is fine. These are the traits that you have. In this case, I have a brown from maybe my mom, and a blue from my dad. In this case, I have a blue from both my mom and dad. How do we know whether my eyes are going to be brown or blue? And the reality is that it's very complex. It's just a whole mixture of things. But Mendel, he studied things that showed what we'll call dominance. And this is the idea that one of these traits dominates the other. So a lot of people originally thought that eye color, especially blue eyes, was always dominated by the other traits. We'll assume that here, but that's a gross oversimplification. So let's say that brown eyes are dominant. Brown are dominant. And blue are recessive. Blue, I wanted to do that in blue. And blue eyes are recessive. If this is the case, and this is as I've said repeatedly, this is a gross oversimplification, but if that is the case, then if I were to inherit this genotype, because brown eyes are dominant, remember I said the big B here represents brown eyes, you won't, and the lowercase b is recessive, all you're going to see for the person with this genotype is brown eyes. So let me do this here. Let me write this here. So genotype, and then I'll write phenotype. And genotype is the actual versions of the gene you have, and then the phenotypes are what's expressed, or what do you see? So phenotype, confused. So if I have a brown-eyed gene from my dad and a blue-eyed gene from my mom, color transitions aren't there. A blue-eyed gene from my mom, because the brown eye is recessive, the brown-eyed allele is recessive. I just said a brown-eyed gene, but what I should say is the brown-eyed version of the gene, which is the brown allele, or the blue-eyed version of the gene from my mom, which is the blue allele. Since the brown allele is dominant, I wrote that up here, what's going to be expressed are brown eyes. Brown eyes. Now, if I have, let's say if I had it the other way, let's say I got a blue-eyed from my dad, and I get a blue-eyed allele from my dad, and I get a brown-eyed allele from my mom. Same thing, the phenotype is going to be brown eyes. Now, what if I get a brown-eyed allele from both my mom and my dad? I keep changing the shade of brown. They're all supposed to be the same. So let's say I get two dominant brown-eyed alleles from my mom and my dad. Then what two types of alleles we might see are going to dominate it. So all of a sudden, the phenotype will be blue eyes. And I want to repeat again, this isn't necessarily how the alleles for eye color work, but it's a nice simplification to maybe understand how heredity works. And there are some traits that can be studied in this simple way. But what I wanted to do here is to show you that many different genotypes, so these are all different genotypes, they all coded for the same phenotype. So just by looking at someone's eye color, you didn't know exactly whether they were homozygous dominant, this would be homozygous dominant, or whether they were heterozygotes. This is heterozygous right here. These two right here are heterozygotes. Heterozygotes. These are also sometimes called as hybrids. But the word hybrid is kind of overloaded. It's used a lot. But in this context, it means that you got different versions of the allele for that gene. So let's think a little bit about what's actually happening when my mom and my dad reproduced. So my dad, well, let's think of a couple of different scenarios. Let's say my dad was, let's say that they're both hybrids. My dad has the brown dominant, and she also has the blue-eyed recessive allele. Now let's think about if these two people, before you see what my eye color is, if you said, look, I'm giving you what these two people's genotypes are. Let me label it. This is the genotypes that their children could have. So let's say they reproduce. Well, I'm going to draw a little grid here. So let me draw a grid. All right. So we know from our study of meiosis that, look, my mom has this gene on, let me draw the genes again. So there's a homologous pair, right? This is one gene right here, one chromosome right here. That's another chromosome right there. On this chromosome in the homologous pair, there might be at the eye color locus, there's the brown eye gene. And at this one, at the eye color locus, there's a blue eye gene. And similarly for my dad, when you look at that same chromosome in his cells, so let me do them like this. So this is one chromosome there, this is the other chromosome here. When you look at that locus on this chromosome, or that location, it has the brown-eyed allele for that gene, and on this one it has the blue-eyed allele on this gene. And we learned from meiosis when the chromosomes, well, they replicate first, and so you have these, two chromatids on a chromosome, but they line up in meiosis 1 during the metaphase, and we don't know which way they line up. For example, my dad might give me this chromosome, or might give me that chromosome, or my mom might give me that chromosome, or might give me that chromosome. So I can have any of these combinations. So for example, if I get this chromosome from my mom and this chromosome from my dad, what is the genotype going to be for eye color? Well, it's going to be capital B and capital B. If I get this chromosome from my mom and this chromosome from my dad, what's it going to be? Well, I'm going to get the big B from my dad, and then I'm going to get the lowercase b from my mom. So this is another possibility. Now, this is another possibility here where I get the brown-eyed allele from my mom, and I get the blue-eye allele from my dad. And then there's a possibility that I get this chromosome from my dad and this chromosome from my mom. So it's this situation. Now, what are the phenotypes going to be? Oh, we've already seen that this one right here is going to be dad and this chromosome from my mom. So it's this situation. Now, what are the phenotypes going to be? Oh, we've already seen that this one right here is this one's going to be brown, that one's going to be brown, this one's going to be brown, but this one is going to be blue. I already showed you this. But if I were to tell you ahead of time that, look, I have two people, they're both hybrids, or they're both heterozygotes for eye color, and eye color has this recessive dominant situation, and they're both heterozygotes where they each have one brown allele and one blue allele, and they're going to have a child. What's the probability that the child has brown eyes? Brown eyes. What's the probability? Well, each of these scenarios are equally likely, right? There's 4 equal scenarios. So let's put that in the denominator. 4 equal scenarios. And how many of those scenarios end up with brown eyes? Well, it's probability that the blue eyes that they produce, which are heterozygous. Well, this one is because it has a mix. It's a hybrid. It has resulted in a heterozygote. Whether one's dominant or one's recessive. We can start to make predictions about the probabilities of. In the last video, I drew of different outcomes, and it was brown eyes, was zygotes or something. It is a bit of oversimplification, but many traits, especially some of the things that Gregor and Mendel studied, can be studied in this way. Gregor and Mendel studied can be studied in this way. Last video, I drew this grid in order to better the different combinations of alleles I could get from my mom or my dad. And this grid that I drew is called a Punnett square. Punnett square. And I looked up what Punnett means. And it turns out, and this might be the biggest takeaway from this video, that when you go to the farmer's market or you go to the produce and you see those little baskets, that often you'll see maybe strawberries or blueberries sitting in. That's a little grid here, right there. Sometimes grapes are in them, and you have a bunch of strawberries in them, like that. That green basket is a Punnett. That's a Punnett. Apparently, in some countries they call it a Punnett. I think England's one of them, and then UK viewers can correct me if I'm wrong. And so I guess that's where the inspiration comes from about calling these Punnett squares. These are kind of these little green baskets that you can throw different combinations of genotypes in. And these Punnett squares aren't just useful if you're talking about crossing 2 hybrids. This is called a monohybrid cross because you are crossing 2 hybrids for only one trait. It could be useful for a whole set of different types of crosses between two reproducing organisms. It doesn't even have to be a situation where one thing is dominating another. Let's do a bunch of these just to make you familiar with the idea. So let's say you have a mom. So instead of doing 2 hybrids, let's say the mom. I'll keep using the blue-eyed, brown-eyed analogy just because we're already reasonably useful to it. Let's say that she's homozygous dominant, and let's say that the dad is a heterozygote, so he's got a brown and he's got a blue, and we want to know the different combinations of genotypes that one of their children might have. So what we do, brown allele, assume. Oh, you write that homozygous dominant, and now we're looking at the genotype. We care about specific alleles that child. I'll break down a little bit with parents. Although there is a male and female sometimes on the same plant, and let's say the other plant is also red and white. So the other plant has a red allele that have two. They don't even have to be. You can have red flowers, or you could have white flowers, or you could have white flowers. Let's say I were to cross a parent flower where that has the genotype capital R. I'll just make it a capital W. So that could be the mom or the dad. Or the analogy breaks down a little bit with parents. Although there is a male and female sometimes on the same plant. And let's say the other plant is also red and white. So the other plant has a red allele and also has a white allele. So what are the different possibilities? Well, we just draw our Punnett square again. Let me draw our little grid. So the child can inherit both of these red alleles. Could inherit this white allele and then this red allele. So this red one and then this white one. Right, that's that right there. And that red one is that right there. Or could inherit this red one from, let's say this is the mom plant, and then the white allele from the dad plant. So that's that one right there. Or you could inherit both white alleles. And what I said when I went into this, and I wrote it at the top right here, is we're studying a situation dealing with incomplete dominance. So what does that mean? Well, that means we might actually have mixing or blending of the traits when you actually look at them. So this was complete dominance. If red was dominant to white, you say, okay, all of these guys are going to be red, and these guys, only this guy right here is going to be white. So you have a 1 in 4 probability of being white. But let's say that a heterozygous genotype. So let me write that down. Let's say that R, you have one R allele and one white allele. And this doesn't result in red. This results in pink. This results in pink. So this is what blending is. It's kind of a mixture of the two. So if I said if these two plants were to reproduce, and the traits for red and white petals, I guess we could say, are incomplete dominant or incompletely dominant, or they blend. And if I were to say, what's the probability of having a pink plant? And now when I'm talking about pink, this of course is a phenotype. So the probability of pink, well, let's look at the different combinations. How many of these are pink? This one is pink, and this is pink. So 2 are pink of a total of 4 equally likely combinations. So it's a 50% chance. It's a 50% chance that we're pink. And we could keep doing this over multiple generations. Say, oh, what happens in the second and the third and the fourth generation? Actually, we could even have a situation where we have multiple different alleles. And I'll use almost a kind of a more realistic example. I'll use blood type as an example. So there are 3 potential alleles for blood type. 3 potential alleles. You can have a blood type A, you can have a blood type B, or you can have a blood type O. You can have a blood type O. And what happens is you have a combination here between codominance and recessive genes. And I'm going to show you what I talked about when we do the Punnett squares. Maybe I'll stick with one color here because you're getting the idea. So let's say I have a parent who is AB. So that means that they have on one of their homologous chromosomes, they have the A allele, and the other one they have the B allele. That's what AB means. So the phenotype is the genotype. They're codominant. They both express themselves. They don't necessarily blend. They both express. That's an AB blood type. So let me write this right here. This is AB blood type. And then the other parent. So the other parent is let's say that they are fully an A blood type. Let's say they're an A blood type. Let's say their phenotype is an A blood type. I hope I'm not confusing you, but their genotype is that they have one allele that's an A and their other allele that's an O. So this is what's interesting about blood types. It's a mixture. O is recessive. While these guys are codominant. So if you have either of these guys with an O, these guys dominate. If you have them together, then your blood type is AB. So what are all the different combinations for this couple here? For this couple right here? Well, you could get this A and that A. So you get an A from your mom, and you get an A from your dad right there. And clearly in this case, you'll have your phenotype. You have an A blood type in this situation. You could get the A from your dad, and you could get the B from your mom, in which case you have an AB blood type. You could get the A from your mom and the O from your dad, in which case you have an A blood type because this dominates that. Or you could get the B from your, don't want to introduce arbitrary colors. You could get the B from your mom, like this way, B from your mom, that's this one, or the O from your dad. And this, no one's going to introduce a different color. And this is a B blood type. So if I said, what's the probability of having an A blood type? Well, and once again, we're talking about a phenotype here. So which of these are an A blood type? Well, this one definitely is. Because it's AA. If you have 2 A alleles, you definitely have an A blood type. But you also have an A blood type phenotype if you have an A and then an O.
All right, let's take the NyQuil. Let's shoot for 11 a day. All right, let's see how far 2 hours gets us.
O is recessive. So this is also going to be an A blood type. So AO, these are both A blood. So there's a 50% chance because 2 of the 4 combinations show us an A blood type. And you can do all the different combinations. You say, well, how do you have an O blood type? Well, at least both of your parents will have to carry at least one O. So for example, to have an A, that would have been possible if maybe instead of an AB, this right here was an O. Then this combination would be like 2 O's right there. So hopefully that gives you an idea of how Punnett square can be useful. It can even be useful when we're talking about more than one trait. So let's go to our situation that I talked about before, where I said,
Isn't that
we
already
have little b
more
is
than one
equal to blue eyes, and we're assuming that that's recessive. And you have big B is equal to brown eyes, and we're assuming that this is dominant. And let's say we have another trait. We call it, well, I introduced that tooth trait before. So let's say little t is equal to small teeth. I don't know what type of bizarre organism I'm talking about, although I think I would fall into the big tooth camp. Let's say big T is equal to big teeth. So an individual can have, for example, I might be heterozygous brown eyes. So my genotype might be heterozygous for brown eyes, and then homozygous dominant for teeth. So this might be my genotype. And the phenotype for this one would be a big tooth, brown-eyed person. Look at before. So let's say little t is equal to small teeth. I don't know what type of bizarre organism I'm talking about, although I think I would fall into the big tooth camp. Let's say big T is equal to big teeth. So an individual can have, for example, I might be heterozygous brown eyes. So my genotype might be heterozygous for brown eyes, and then homozygous dominant for teeth. So this might be my genotype. And the phenotype for this one would be a big tooth, brown-eyed person, right? Let me make that clear. This is big tooth phenotype, big tooth, and this is the phenotype. What you see is brown eyes, a big tooth, brown-eyed person. Now, if we assume that the genes that code for teeth or eye color are on different chromosomes, and this is a key assumption, we can say that they assort independently. And this is the phenotype. And this is the phenotype. What you see is brown eyes, a big tooth, brown-eyed person. Now, if we assume that the genes that code for teeth or eye color are on different chromosomes, so this is a key assumption, we can say that they assort independently. Let me write that down. Independent assortment. Assortment. So this is a case where if I were to look into look at my chromosomes, let's say this is one homologous pair, maybe we call this homologous pair 1. And let's have another homologous pair. Obviously we have 23 of these, but let's say this is homologous pair 2 right here. If the eye color gene is here and here, remember the both homologous chromosomes code for the same genes. They might have different versions, those are alleles. And if teeth are over here, obviously we have 23 of these, but let's say this is homologous pair 2 right here. If the eye color gene is here and here, remember the both homologous chromosomes code for the same genes. They might have different versions, those are alleles. And if teeth are over here, they will assort independently. So when after meiosis occurs to produce the gametes, the offspring might get this chromosome or copy of that chromosome for eye color, and might get a copy of this chromosome for teeth size or tooth size. Or it could go the other way. Maybe it's another offspring gets this one and this chromosome for eye color, and then this chromosome for teeth color, and gets the other version of the allele. So because they're on different chromosomes, there's no linkage between whether if you inherit this one, whether you inherit big teeth, whether you inherit small brown eyes or blue eyes. Now, if they were on the same chromosome, let's say the situation where they are on the same chromosome. So let's say we pick another trait, hair color. Let's say the gene for hair color is on chromosome 1. So let's say hair color, the gene is there and there. These might be different versions of hair color, different alleles, but the genes are on that same chromosome. In this situation, if someone gets, let's say if this is blue eyes here, and this is blonde hair, then these are going to always travel together. You're not going to have these assort independently. These are called linked traits. Let me highlight that. So these right there, those are linked traits. But for a second, we'll talk more about linked traits, and especially sex-linked traits in probably the next video or a few videos from now. But let's assume that we're talking about traits that assort independently. And we cross 2 hybrids. So this is called a dihybrid cross, very fancy word, but just to give you an idea of the power of the Punnett square. So let's say both parents they're both hybrids, which means that they're both have the dominant brown eye allele, and they have the recessive blue eye allele, and they both have the dominant big tooth gene, and they both have the recessive little tooth gene. So this is the genotype for both parents. Both parents are dihybrids. The hybrids for both genes, both parents. What are all the different combinations for their children? And I could have done this without dihybrids. I could have made one of them homozygous for one of the traits and a hybrid for the other, and I could have done every different combination. But I'll do the dihybrids because at least a lot of variety. You'll often see this in classes. So if I'm talking about the mom, what are the different combinations of genes that the mom can contribute? Well, the mom could contribute the brown. So for each of these traits, she can only contribute one of the alleles. So she could contribute this brown right here, and then the big yellow T. So this is one combination. Or she could contribute the big brown, and then the little yellow T. Or she can contribute the blue eye allele and the big T. So these are all the different combinations that she can contribute. And then the final combination is this allele and that allele. So the blue eyes, and the small teeth. So that's for mom. And of course, dad can contribute the same different combinations because dad has the same genotype. Let me write that down. So brown eye, let me just write it like this so I don't have to keep switching colors. I think I want to make them a little closer together because I'm going to run out of space otherwise. Nope. Let me do it like that. Okay. Brown eye. So the dad can contribute the big the big teeth or the little teeth along with the brown eye gene, or if you contribute the blue eye gene, or the blue eye allele, and in combination with the big teeth or the yellow teeth, not the yellow teeth, the little teeth. That would be a different gene for yellow teeth. I mean, that's an environmental factor. So these are all the different combinations that can occur for their offspring. So let's call this maybe a super Punnett square, because we're now dealing instead of 4 combinations, we have 16 combinations. 16 combinations. Looks like I ran out of ink right there. Right there. It's strange. Why 16 combinations? Write that out. Something's wrong with my tablet. Maybe there's something weird. Okay, so there's 16 different combinations, and let's write them all out. And I'll just stay in one, maybe neutral color, so I don't have to keep switching. I could get this combination. So this big brown eyes from my mom, brown eyes from my dad allele. So it's brown, and then big teeth from both. I could have this combination. So I have capital B and a capital B, and then I have a capital T and a lowercase t. And then let's just keep moving forward. So I get a capital B and a lowercase b with a capital T and a capital T, a big B, lowercase b, capital T, lowercase t. And I'm just going to go through these super fast. It's going to take forever. So capital B from here. For example, how many of these are going to exhibit brown eyes and big teeth? So big teeth, brown-eyed kids, big teeth. Big teeth. Let me write this down here. So if I want big teeth, and brown eyes. Oh, my pen doesn't. Brown eyes. So how many are there? Big teeth and brown eyes. So we have to just, they're both the dominant. So if you have either a capital B or a capital T in any of them, you're going to have big teeth and brown eyes. So this is big teeth and brown eyes. This is big teeth right here, brown eyes there, big teeth. I'm going to say brown eyes and big teeth, because that's the order that I wrote right here. Brown eyes and big teeth, brown eyes and big teeth. Even though I have a recessive trait here, the brown eyes dominate, and I have a small teeth here, but the big teeth dominate. This is brown eyes and big teeth. This is brown eyes and big teeth. Let's see. This is brown eyes and big teeth, brown eyes and big teeth. And let me see is that all of them? Well, no, this is brown eyes, little teeth. This is brown eyes and big teeth right there. And this is also brown eyes and big teeth. They're heterozygotes for each trait, but both brown eyes and big teeth are dominant. So these are all phenotypes of brown eyes and big teeth. So how many of those do we have? We have 1, 2, 3, 4, 5, 6, 7, 8, 9 of those. So we have 9 brown eyes and big teeth. Now, how many do we have of big teeth? So let me write teeth. What are the combinations there? Well, this is blue eyes and big teeth, blue eyes and big teeth, blue eyes and big teeth. So there's 3 combinations there. And if I want to be recessive on both traits, so if I want, let me do this. I want blue eyes, blue, and little teeth. Little teeth. There's only one. Out of the 16, there's only one situation where I inherit the recessive trait from both parents for both traits. So if you look at this and you say, hey, what's the probability? There's only one of that. What's the probability of having a big teeth, brown-eyed child? And these are all the phenotypes. There are 16 different possibilities here, right? There's 16 squares here. 9 of them describe the phenotype of big teeth and brown eyes. So there's a 9/16 chance. So it's 9 out of 16 chance of having a big teeth, brown-eyed child. What's the probability of a blue-eyed child with little teeth? 1 in 16. 1 in 16. So hopefully in this video you've appreciated the power of the Punnett square. This is a useful way to explore every different combination of all.
Sure.
What you see depicted here is known as a pedigree. And a pedigree is a way of analyzing the inheritance patterns of a trait within a family. And it can be useful to understand more about that trait, maybe to make some insights about the genetics of that trait. And it's a way to think about, well, what's happened in the past in the family, and then maybe we can help get some probabilities or get some understanding of what might happen in the future. But let's just break it down so that we understand all of the different parts of a pedigree. So big picture, a circle represents a female in the family, and a square represents a male in the family. If either the circle or the square is colored in, like we see right over here, or we see right over here, that means that that person exhibits the trait. And the trait that this particular pedigree is for is the trait of freckles. And whether or not we're colored in, that is a phenotype. That is an observed trait. You just look at the person and say, do they have freckles or not? This male right over here does not have freckles. This female right over here does have freckles. This male right over here does have freckles. But once again, that's phenotype. That's the observed characteristic. We don't know exactly the genotype just from this, although we might be able to infer some ideas about the genotype by seeing how the phenotype pattern spread from generation to generation. But just going back to understanding the pedigree itself, when you have a horizontal line connecting a male and a female, it's called a marriage line. And it means that they are coupling up and they are reproducing. They don't necessarily, I guess, have to be married. It's more that they reproduce. So this male and this female have reproduced. And then this vertical line that goes from that horizontal line, that's known as the line of descent. So it's going from that first generation to the second generation. And so all of the people connected to that vertical line at the bottom of the vertical line, these are their children, all of the people who are directly connected to this. And this is known as a sibling line. So this person, and that person, they are all brothers and sisters. You can see that generation 1, they had 2 daughters, one of whom exhibited freckles, and 1 son who had freckles. Now, these other people in generation 2, these aren't children of the first generation. These are people, you could say, who were brought into the family. They either married into the family, or you could just say they reproduced with the children. But that's what's happening with generation 2. And so you can see you have another what's known as a marriage line, but I guess it could be a coupling line, a line of descent. And then they have 2 daughters in this case. So it turns out that freckles is an autosomal dominant trait. And I will explain what that means in a second. Autosomal dominant trait. Autosomal means that it is a trait associated with one of the non-sex chromosomes. We have 23 pairs of chromosomes as humans. One of those pairs determines our sex. The other ones don't determine our sex. So this means that freckles is associated with one of the 22 pairs of chromosomes that do not determine your sex. And dominant means that in general, you get 2 versions of a gene, one from your mom and one from your father. And each of those versions of the gene for, say, whether or not you have freckles is known as an allele. And if a trait is autosomal dominant, that means that if you have even just one allele for that trait is known as an allele. And if and one from your father. And each of those versions of the gene for, say, whether or not you have freckles is known as an allele. And if a trait is autosomal dominant, that means that if you have even just one allele for that trait, that you are going to exhibit the trait. And so, for example, if the alleles for freckles, if capital F is you have freckles, it's dominant, so we write in capital, and lowercase f means that you don't have freckles. If someone has a genotype of capital F, or capital F, lowercase f, or I guess we'd say lowercase f, capital F, if we wanted to think about order, which one you're getting from which parent. Well, then in all of these situations, they would exhibit the phenotype of freckles. We talk about genotype and phenotype in other videos. And of course, the only genotype in which they would not exhibit freckles would be having both of the recessive traits. So with this out of the way, what can we infer about the genotype of this individual right over here? And this individual will sometimes be referred to as individual 1, 2. They're in generation 1, and they're individual 2 in generation 1. Well, we know that they exhibit freckles. So we know for sure that since the phenotype is freckles, they're in one of these scenarios. And these two are frankly equivalent. So this person, we know that they're either capital F, so that would be homozygous dominant, or they're capital F, lowercase f, which are they are heterozygous. Well, let's first think about the situation if they were homozygous dominant. If they're homozygous dominant, that means to every one of her children, she would give one of these alleles, and either of those are dominant. And so that would mean, if this were the case, that all of her children would get at least one dominant allele. Well, if they all get at least one dominant allele, then all of her
That
children
means 2 would show. So she's
would
heterozygous.
have freckles. But we're seeing a situation where all of her children do not have freckles. 2 of her 3 children had freckles. So this cannot be the case. So this individual right over here must be heterozygous for freckles. So they must be capital F, lowercase f. And what do we know about this individual right over here, the person that she coupled up with? Well, they don't exhibit the phenotype of freckles. So they must be lowercase f, lowercase f. And then from that, you might be able to input a lot more about that. We'll do that in future work examples.
Oh.
Try this cherry splash.
No.
Ya. Te tomaste una Sprite.
No puedo, pero es diferente.
La misma cosa.
No, es diferente, mamá.
Zero sugar.
No, es diferente.
Aquí tuviera la maquela, te la dieras here, drink it. Y te la tomas.
All right. Let's continue on with this pedigree stuff. I got some food. Thank the gods. That's funny. Oh, I see. But I do not influence our color or see shape. Hmm. Hmm. The goat. Okay, I think that's enough. Like a little background. I think I was here. The law of independent assortment. Independently of one another. Gotcha. Right. Those are all round. Didn't follow the independent assortment. Gotcha. Gotcha. If the two genes are very close together. Combo unit. Two copies of a gene. Homologous. 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 Metaphase 1. Uh, the very far part. So black is dominant. Yellow and straight fur. Well, then it's going to be the 16. And since this is the recessive side, I think we go with straight. Oh, I think we're going to go with 116, right? Oh, then 316. Oops. BB yellow. CC is straight. Okay. It's going to be straight. Sure. I said there's only one question. I guess so. Incomplete dominance. Incomplete dominance. Gotcha. RR, RW, RW, WW. It's both red and white. How many can I take? An equal number. Oh my goodness. Chinchilla. It's a chinchilla. Himalayan. Albino. A defective enzyme. Are you sensitive to temperature? Oh. High temperature? Doesn't work at all high temperature. No pigment is made. Non-functional enzyme. Hmm. Two videos and three. Excuse me. 2 videos and 3 articles. Dang, only got to 2 today. Almost used to be the limit, but I don't think so. I think I could make 4 if I wake up earlier. Anyways, I mean, this one's pretty short, so. Oh man, that means I'm gonna need to go behind. Oh, we'll see. Anyways, let's keep going.
Incomplete dominance. But first, let's review the example of blood type. So today we're going to talk about codominance and incomplete dominance. But first, let's review the example of blood type and how someone with the same 2 alleles coding for the same trait would be called homozygous, and someone with different alleles would be called heterozygous.
Right.
Also remember the concept of dominant and recessive alleles, and how the A allele is dominant over the O allele in this example. This means that the same phenotype, blood type A, can result from these 2 different genotypes. Now, the example that I just gave you was an example of complete dominance. So if the person had a genotype AO, since our phenotype is just blood type A, it means that the A allele is completely dominant over the O allele, and only the A allele from the genotype is expressed in the phenotype. But there are actually 3 different patterns of dominance that I want you to be familiar with. And to explain this, I'm going to use a different example. So let's say we have this flower, and the red petal phenotype is coded for by the red R allele, and the blue flower phenotype is coded for by the blue R allele. So I'm going to introduce 3 different patterns of dominance, and they are complete dominance, which you've already heard of.
Let me wash my hands real quick.
Codominance and also incomplete dominance. And I'm going to explain what these 2 new patterns are through this flower example. So let's start by looking at 3 different genotypes and the phenotypes that you see for each of them under each different dominance pattern. So we'll start with the genotype 2 red Rs, which we can expect that in all cases the flower petals will be red, since we only have red Rs in the genotype. Similarly, if our genotype had 2 blue Rs, then we can expect that in all complete dominance. And I'm going to explain what these two coding for the same trait would be called homozygous, and someone with different alleles would be called heterozygous. Also remember the concept of dominant and recessive alleles, and how the A allele is dominant over the O allele in this example. This means that the same phenotype, blood type A, can result from these two different genotypes. Now, the example that I just gave you was an example of complete dominance. So if a person had a genotype AO, since our phenotype is just blood type A, it means that the A allele is completely dominant over the O allele, and only the A allele from the genotype is expressed in the phenotype. But there are actually 3 different patterns of dominance that I want you to be familiar with. And to explain this, I'm going to use a different example. So let's say we have this flower, and the red petal phenotype is coded for by the red R allele, and the blue flower phenotype is coded for by the blue R allele. So I'm going to introduce 3 different patterns of dominance, and they are complete dominance, which you've already heard of, codominance, and also incomplete dominance. And I'm going to explain what these 2 new patterns are through this flower example. So let's start by looking at 3 different genotypes and the phenotypes that you would see for each of them under each different.
What is this? High school? No.
Dominance pattern. So we'll start with the genotype 2 red Rs, which we can expect that in all cases the flower petals will be red, since we only have red Rs in the genotype. Similarly, if our genotype had 2 blue Rs, then we can expect that in all cases the flower petals will be blue, since we only have blue Rs in the genotype. Now these 3 different dominance patterns change when we look at the heterozygous example, and that's what makes these 3 patterns different. Now, we're already familiar with the example of complete dominance. So if we said that the red R is dominant over the blue R, then this would make the heterozygous phenotype a red flower for complete dominance. Now, what codominance is when the heterozygous phenotype shows a flower with some red petals and some blue petals. So it's when the 2 alleles are dominant together. They are codominant, and traits of both alleles show up in the phenotype. Now, what incomplete dominance is when the heterozygous phenotype shows a mixture of the 2 alleles. So in this case, the red and blue flower petals may combine to form a purple flower.
Ooh.
Neither allele is completely dominant over the other, and instead, the 2 being incompletely dominant mix together. So what did we learn? Well, if we assume the heterozygous genotype, red R, blue R, then there are 3 different dominance patterns that we might see for a specific trait. In complete dominance, only one allele in the genotype, the dominant allele, is seen in the phenotype. And this was the example with the red flower. In codominance, both alleles in the genotype are seen in the phenotype. And this was the example with the flower with both red and blue petals. And finally, in incomplete dominance, a mixture of the alleles in the genotype is seen in the phenotype. And this was the example with the purple flower.
You can explain it, buddy. Oh, you just explain it.
Different combination of all of the genes. It doesn't have to be only one trait. It can be in this case where you're doing 2 traits that show dominance, but they sort independently because they're on different chromosomes. You could use it, where did I do it over here? You could use it to explore incomplete dominance when there's blending, where red and white make pink genes, or you can even use it when there's co-dominance and when you have multiple alleles, where it's not just 2 different versions of the gene, there's actually 3 different versions. So hopefully you've enjoyed that.
Sure. Maybe we can just take a break. The rest of these I'm just gonna read over. So let's just take let's take a 5-minute. I'll take a 5-minute. I'll do this one and this one, which they seem to be actually different. Well, this one has fruit flies. This one has some of it. Some genetic linkage stuff. I don't know. I think I would do this one first and then this one. And we'll add to Anki today. I don't think we'll do high yield. Well, we might. We'll read high yield in the morning as soon as I wake up for these 2, and then yeah, we'll continue. Eh? We shall continue on. Oh, okay. Oh, nice. That's not that bad. I think I can make it. 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 Luckily, we haven't hit this yet, so that's on week 4. So, yeah, we're on week 2, but we'll see if I can catch up. Because there's a little bit less sections on this then. Oh my god, I'm talking too much. Shut up. 1, 2, 3, 4, 5, 6, 7, 9, 10, 11. These are the same. 3, 4, 5, 6, 7, 8, 9, 10. 2, 3, 6, 7, 8, 9, 10, 11. Oh, it's the same. It's actually the same. I don't know if I can do that, but I'm gonna try. Okay. Oh, this is not working. Yeah, okay, I can do it. I can do it. I can do it. I'm gonna try. I'm gonna try. Why is this still here like this? It's weird. I don't know. Whatever. Yeah, that's a good question. Hmm. 1/2 times 1/2. Well, that's not too bad. I need to study. Okay. Okay. Did I use it for this one? No, I guess not. No. I think. Oh, I see. So it's always good. Ah. I think it's gonna be. Oh man, 1/16. Oh, whoops. I messed up. That actually is what I got. Oh, what? 0? Why is 0? Oh, 4 times 1/2 times 0 times 1/2. Oh, okay. Oh, because the other one doesn't have it. Oh, gotcha. Okay. Sure. Let's let's add this. Actually, All right, guys. Thanks for watching. Have a good night.